हेलो स्टूडेंट्स, कंफर्म मी वंस इफ आई एम ऑडिबल एंड वीडियो इज क्लियर। अगर वीडियो ऑडियो दोनों क्लियर है तो एक बार चैट बॉक्स में थम्स अप करना। इफ ऑडियो एंड वीडियो बोथ आर क्लियर, okay. So hello everyone, welcome back to our YouTube channel AgriAddict, and this is the last session of the marathon series for ICAR PG exam 2026 for the subject of genetics. Don't worry, this is the last marathon for the subject genetics. ठीक है, genetics के लिए last marathon है, total overall last marathon नहीं है. And now in today's class, so we are going to continue our discussion on the molecular genetics. Molecular genetics जो हमने लास्ट क्लास में डिस्कस किया था, स्टार्टिंग के कुछ पार्ट्स को कंटिन्यू करेंगे और मॉलिक्यूलर जेनेटिक्स को खत्म करेंगे, ताकि आपके एग्जाम में इन योर एग्जाम लाइक व्हाटएवर क्वेश्चंस दैट यू विल बी गोइंग टू अटेंड, एवरीथिंग विल बी क्लियर फ्रॉम दिस मैराथन सीरीज. ठीक है, ओके. एंड सर, हाउ मच इज दिस स्कोर? जैसा हम थिंक व्हाट मॉक टेस्ट, सिद्धार्थ शर्मा. अच्छा सर, कितना स्कोर है जिससे हम सेफ समझे इस मॉक टेस्ट में? से इफ यू आर स्कोरिंग एनीवेयर अबव 300 प्लस, अगर आप 300 प्लस स्कोर कर पा रहे हो इन मॉक टेस्ट में, सो इट्स वेल एंड गुड, इट्स वेरी गुड दैट इफ यू आर स्कोरिंग इट. एंड अगर आप 350 प्लस स्कोर कर रहे हो देन इट्स बेटर, एंड इफ बाय एनी चांस इफ यू आर स्कोरिंग मोर देन 400 इट्स बेस्ट. इतना मैं बोलना चाहूंगा. एंड ऑल्सो लाइक इफ यू आर टारगेटिंग फॉर गेटिंग द ICAR PG फेलोशिप, अगर आप PG फेलोशिप के लिए टारगेट कर रहे हो, तो इन द मेन एग्जाम ऑल्सो अगर समवेयर और द अराउंड लाइक आपने अगर 330 प्लस स्कोर कर लिया देन फॉर श्योर लाइक यू विल गेट द JRF. ओके, तो इतना तो श्योर है. बट अगर जेनेटिक्स में ही JRF फेलोशिप चाहिए या प्लांट पैथोलॉजी में ही चाहिए तो देन यू हैव टू सिक्योर लाइक 350 प्लस तो एग्जाम में आना चाहिए. इफ द पेपर लेवल इज सेम और सिमिलर टू व्हाट द पेपर इज फ्रॉम द डिफरेंट ईयर्स, लाइक जैसे कि 2020 से लेकर 2024 तक जैसा पेपर का लेवल था ऐसा ही अगर रहेगा 2026 का भी, तो यू कैन एक्सपेक्ट दिस. ठीक है, आई स्कोर 284 बट ओके. सो अगर आप लोग लाइक 300 से नीचे स्कोर कर रहे हो सो डोंट वरी, लाइक जस्ट पुश योरसेल्फ. जो वीक एरियाज है उसको आइडेंटिफाई कर लो. इन 3 दिनों में उसके लिए क्या कर सकते हो उसको सोचो. और अगर सिली मिस्टेक की वजह से आपने 300 से नीचे आपने स्कोर किया है तो यू हैव टू प्रैक्टिस करने के लिए ज्यादा टाइम नहीं है. बट अगर you are at least practice like 1 or 2 mock tests and try to push yourself more than 300, that will definitely build the confidence. उससे तो कॉन्फिडेंस आ ही जाएगा. यस सर, मेरा 15 सिली मिस्टेक वेरी इजी क्वेश्चन था, फिर भी ऑल आई करेक्ट 336. गुड गुड. वही है, सिली मिस्टेक जिनका भी हुआ है ना, आप लोगों के पास एक ऑप्शन ये है कि और 2-3 मॉक टेस्ट, मिनिमम तो 2-3 मॉक टेस्ट आपने अभी तक अटेंड करके नहीं रखा होगा. जो नहीं किया है अटेंड, जो टेस्ट सीरीज में ऑलरेडी अपलोडेड है उसको अच्छे से अटेंड कर लेना. पुश योरसेल्फ, ताकि उसमें आप एटलीस्ट सिली मिस्टेक्स नहीं करो. एंड ऑल्सो लाइक पुश योरसेल्फ इफ यू आर सिक्योर्ड लाइक लेस देन 300 टू ओके मेक इट ओवर इट. यस ठीक है. ओके, लेट्स कीप दिस डिस्कशन एट द एंड. लास्ट में डिस्कशन को एंड रखेंगे. एंड इन दिस टुडेस क्लास वी आर गोइंग टू डिस्कस एंड कंटिन्यू द डिस्कशन ऑन द मॉलिक्यूलर जेनेटिक्स. ठीक है, एंड आई एम गोना मिस यू ऑल. मिस करूंगा, ठीक है, आप सब लोगों को. आई विल मिस यू सो मच, but still hope so like even after the exam one fine day we'll meet face to face. And if you are securing top rank and you will be coming into IRI, then definitely we will be eagerly waiting to welcome you all. And not just IRI, we will be coming to every top agriculture universities in the next year. I would be very happy that if I am able to meet you there and you just recognize yourself as a student of AgriAddict and recognize yourself as a student of the master course as a test series. So I'll be the most happiest person on the earth. Okay. seed sense marathon करेंगे. That's what I told you. This is the last marathon of genetics, but after this we'll also have the one session of seed sense. ठीक है, देखो seed tech का मैसेज आ ही रहा है. I think like मार दोगे अगर मैं बोलूंगा कि seed tech का नहीं करेंगे तो मार दोगे मुझे लगता है. Okay, okay sir. Okay guys, so let's start. So yesterday we discussed till the RNA processing. RNA processing तक डिस्कस किए थे. तो आज हम क्या डिस्कस करने वाले हैं, हम उसके आगे डिस्कस करने वाले हैं. एक अगर आउटलाइन दे दूं. So in today's class, what we are going to discuss, like we are going to discuss about genetic code. पहला हम genetic code डिस्कस करेंगे. किसी ने लास्ट वीडियो में कमेंट किया था कि थोड़ा अच्छे से हैंडराइटिंग सुधार लो, कि अच्छे से लिख लो, तो आई ट्राई माय बेस्ट कि मैं अच्छा लिख लूं. मेरा हैंडराइटिंग ऐसा तो अच्छा है बट बोर्ड में इतना अच्छा नहीं आता है. नेक्स्ट we'll discuss the genetic code. After that we'll discuss the translation. उसके बाद we'll discuss about the regulation of gene action. Regulation of gene action both in prokaryotes and eukaryotes. Next, so we will discuss about the transposable elements. So this will complete our entire molecular genetics. ठीक है, इससे पहले हमने क्या क्या डिस्कस किया था. इफ यू रिमेंबर फ्रॉम द मॉलिक्यूलर जेनेटिक्स, आपको ऐसा ही याद रखना होगा. फर्स्ट वन इज लाइक जेनेटिक मटेरियल, सेकंड वन इज ऑर्गेनाइजेशन ऑफ जेनेटिक मटेरियल, एंड नेक्स्ट वी डिस्कस्ड अबाउट जीन एक्शन 1 and we discussed gene action 2, and then we discussed like now we have to discuss like gene action 3 which is translation. बस इतना ही है यार. like this is the outline for the molecular genetics. molecular genetics में इतना ही है. and इसके अलावा और कुछ chapters है like molecular techniques in biology. molecular techniques in biology इसको आप completely skip कर सकते हो. and there is one more that is like genome maps. इसको भी आप skip कर सकते हो. इसमें और एक चीज अगर ऐड करना चाहोगे तो ऑर्गेनाइजेशन तो ऐड कर ही दिया. ठीक है, बस इन दोनों चैप्टर्स को कंप्लीटली स्किप कर सकते हो. इसमें कंप्लीट स्किप तो कर सकते हो लेकिन थोड़ा कुछ तो पता होना चाहिए. लाइक अबाउट द डीएनए मार्कर्स, एक क्लास हुआ होगा प्लांट ब्रीडिंग में, जितना उसमें बताया गया है लाइक यू हैव टू नो दैट थिंग्स. ठीक है, यस दैट्स इट. अदर देन दैट यू कैन स्किप ऑल द रेस्ट ऑफ द थिंग्स. ओके, सो लेट्स गेट स्टार्टेड. स्टार्ट करें. या so we'll start with the genetic code. genetic code क्या है? first of all, what is genetic code? genetic code is nothing but amino acid language. जो amino acid है उसका language है genetic code. then what is the language of nucleotide? the language of nucleotide is nitrogen bases. it is the language of nucleotides. जो nucleotides है उसका language nitrogen base है which is like ATGC. similarly amino acid का जो language है वो genetic code बोलते हैं. so it is in the form of codons. ठीक है, it is in the form of codons. so first thing, like how we came to know about the genetic code, like what are the different codons that code for the amino acids. so what are the important events? first thing, like we have to understand about what is codon. codon is nothing but a number or a sequence which will code for one amino acids. for example, we have 20 standard amino acids. so whatever might be the sequence that can give us like one single amino acid. let's take the example of methionine. if something is giving methionine, that is our genetic code. That is our codon. okay, so now we already know that the methionine is coded by the 3-letter codon which is AUG. so overall including all these 3 letters collectively it is called as codon. students confuse हो जाते हैं कि codon एक लेटर का मीनिंग बोलते हैं या 3 लेटर को मिला के codon बोलते हैं. तो याद रखना कि तीनों लेटर को मिला के एक स्पेसिफिक टर्म है codon that will code for a specific amino acid. Here in this case it is methionine. ठीक है, AUG code for methionine. We all know that. That is what the codon is. And next, for the first time it was in the 1954. It was in the 1954. The Gamow suggested that the codon have 3 bases. यहाँ पे भी just like in the DNA structure में जैसा problems थे कि helical in nature है, linear है, 2 strand है, 3 stranded है, multiple strands है, ऐसा ही same doubt ना codon के लिए भी आ रहा था कि codon कितने letters के होंगे. तो he was the first one to just propose that it is made up of 3 bases, but उसने एक गलती किया है इसमें. he said like they are overlapping. codons are overlapping. that means like if you take any amino acid, sorry, if you take any of this mRNA sequence, if any of this mRNA sequence, so इसने ये बोला कि जब mRNA sequence is translated into amino acids, so there is a overlapping. this codes for amino acid 1 and this codes for amino acid 2 and next this codes for amino acid 3. ऐसा ओवरलैप कर करके अमीनो एसिड प्रोड्यूस करेगा. बट नाउ वी नो So this clearly gives amino acid 1, next 3 gives amino acid 2, next 3 gives amino acid 4. तो ये अभी हमें पता है, and this is why we call it as like non-overlapping. This is a very important character of the genetic code. Genetic code का एक important character है that the sequences are non-overlapping. Non-overlapping का मतलब यही है कि ये information जब read करेंगे ये overlap नहीं होता है, ठीक है. Next, it in the non-overlapping system, just opposite to it, like what I discussed. So this is what non-overlapping means. That was ultimately proved. Next, with the genetic studies. Next, with the genetic studies in the T4 phase of E. coli. याद रखना, T4 phase of E. coli पूरा genetic code को decode किया गया या उसका सारे information को निकाला गया. And it was by the studies of this T4 bacteriophage in E. coli with the main scientist Francis Crick and the other co-workers. Finally, we got to know about the important characteristics of the genetic code. That is, like the codon is made up of 3 bases and it is made up of non-overlapping sequence. It is degenerate, it is commaless. See, remember this 4 different or 4 important characters of genetic code. इसके अलावा भी बहुत ही important characters है, उसको भी मैं बताऊंगा. So these characters are very important. Exam में directly पूछेंगे कि characteristic के बारे में. If they are not asking a direct question, they will definitely ask about the degenerate. Degenerate को expand करके एक 2 statement में दे देंगे. Like they will give something about degeneracy. Degeneracy भी बोलते हैं या degenerate भी बोलते हैं. So they will give some statements regarding this and they will twist the questions and ask you definitely. So आपको इन सारे का meaning पता होना चाहिए. Let me discuss one by one. Next, for ये तो 4 character के बारे में पता लग गया because of the work on the T4 bacteriophage of E. coli. तो अगर जब भी E. coli का T4 bacteriophage आ जाता है तो एटलीस्ट 2 चीजें आप सब लोगों को याद होना चाहिए. What is it? See, first thing is like it was utilized to understand the characteristics of genetic code. And what is the other thing? Can you please remember and tell me? इसके अलावा और क्या important experiment हुआ था with the help of like T4 bacteriophage? So what was understood and what was it used for? Yes, one amino acid is coded by more than one codon. It is degeneracy. Yes, very good. Codon is triplet by Crick. Codon is triplet by Crick ऐसा है कि पहले गैमो ने इसको सजेस्ट कर दिया था, उसके बाद उन्होंने कंक्लूसिव सारे अलग अलग साइंटिस्ट का वर्क को कंपाइल करके उसको प्रूफ को अक्यूमुलेट करके ये चीजें प्रपोज किया गया. सो क्रेडिट तो एक को नहीं दे सकते, बट दिस इज हाउ द सीरीज ऑफ इवेंट हैव टेकन प्लेस. यस, इट वाज लाइक फाइन स्ट्रक्चर एनालिसिस ऑफ जीन. So the T4 bacteriophage was also an experimental material in the Benzer, the famous Benzer experiment, which gave us the fine structure analysis of a gene. And remember, इससे हमें सिर्फ fine structure analysis का gene का नहीं मिला था. We got a clear understanding about gene. जीन का स्ट्रक्चर एंड फंक्शनल नेचर भी पता चला था हमें कि ये 3 इंपॉर्टेंट कंपोनेंट से बना हुआ है, सिस्ट्रॉन, म्यूटॉन एंड रिकॉन. ये कैसे पता चला? इट इज बिकॉज ऑफ मेकिंग द फाइन स्ट्रक्चर. पूरा डिटेल में एनालिसिस करने के बाद, कौन सा एनालिसिस? लाइक R2 लोकस. इन द T4 bacteriophage analysis of the R2 locus, like we got the complete information regarding the behavior of the gene, the characteristics of a gene. Okay, yes, Benzer experiment. Yes, very good. ठीक है. Next, so going further, like from the studies of Nierenberg, याद रखना कि Nierenberg and Matthew and Enrich Matthew, they were the first one to decode some of the very important and very easy, very simple amino acids with the codons. This was the first codon to be decoded to be having the information of the amino acid, which is phenylalanine. ये first था. उसके बाद lysine और proline था. Like these are some of the very simple codons that meaning were decoded, and it was by Nierenberg and Enrich Matthew. And next it was by Ocha and co-workers. The other total of abhi pata, like now tell me, like if the nitrogen bases are 4 and there are 3 possibilities for every single amino acids, then what are the total possible number of amino acids, different combinations that are possible in the amino acids? कितना different possible combination बन सकता है? Yes, it is by the formula. क्या formula है? It is 4 power n. और n क्यों? n के जगह में 3 होगा क्योंकि like there are 3 places here. So it is 4 into 4 into 4. It is like 64 possibilities are possible. So that means like there are 64 codons overall. ठीक है? So now the 64 codons में 3 कोडन का मीनिंग पता चल गया हमें कि पहली बार कि इट वाज लाइक फिनाइलएलानाइन फॉर ट्रिपल यू एंड लाइसिन फॉर ट्रिपल ए एंड प्रोलिन फॉर ट्रिपल सी. उसके बाद जो बचे हुए कोडॉन्स थे उसका मीनिंग भी उन्होंने डिकोड किया था, एंड The OCHA and co-workers played a significant role in this. And next, the 3 codons UAA, UGA and UAG are the nonsense codons. Uska bhi meaning हमें समझ में आ गया था. Next thing, next for except and ये भी देखो कि characteristics में except for lysine, serine and arginine, the first 2 letters read from the 5 prime to 3 prime end on the mRNA are common for codons of a single amino acid. Single amino acids के लिए common है except these 3. ठीक है. The first 2 letters for others are very common. तो ये भी आप याद कर सकते हो. Okay. Next, the properties of the genetic code. तो यहाँ पे मैंने एक चीज और मिस किया है, मैं बाद में बताऊंगा. Now here who are the main contributors, अगर मैं पूछूं like who are the main contributors for this decoding the genetic code को पूरा समझने में किसका contribution major था, तो it is Nirenberg and Matty, OCHA. But other than this, can you tell me like who are the other scientists जिसको मैंने यहाँ पे mention नहीं किया है, जिनका contribution बहुत ज्यादा है इसमें? Yes, it is Nirenberg तो है ही यहाँ पे. I'll ask this question later. ठीक है, इसको याद रखने यहाँ से. मेरा और एक साइंटिस्ट कंट्रीब्यूशन जब खत्म होगा उसके बाद मैं इसका कंक्लूजन दूंगा. ठीक है. यस, हरगोविंद खुराना गुड. हरगोविंद खुराना गुड. यस, देयर इज वन मोर साइंटिस्ट. वंस आई कम टू दैट स्लाइड आई विल एक्सप्लेन अबाउट दिस. याद रखना. नेक्स्ट, प्रॉपर्टीज ऑफ जेनेटिक कोड. जेनेटिक कोड का प्रॉपर्टी पता होना चाहिए. फर्स्ट इज ट्रिपलेट. दैट मींस the codon is having like 3 base pair combination, 3 base combination. Next it is degenerate. तो degenerate का मीनिंग क्या है? degenerate का मीनिंग इतना सिंपल है कि सिंगल अमीनो एसिड होंगे तो it is coded by more than one codon. ऐसे भी अमीनो एसिड्स है which are coded by more than one codon. For example, बहुत ही सिंपल एग्जांपल ले लो, phenyl alanine. Phenyl alanine, we know it right now. It is coded by U and U. Okay, एक तो A हो गया. This is like, let's say like this is a codon 1. And there are other codon like UUC. This is a codon 2 and this also codes for phenyl alanine. जहाँ पे देख पा रहे हो कि दो अलग-अलग codons है जो same amino acid को या single amino acid को code कर रहा है. And this behavior is called as degenerate or it is also called as degeneracy. Confuse मत हो जाना, एग्जाम में दोनों सेम ही है, degeneracy या degenerate. ठीक है. Next, it is unambiguous in nature. Unambiguous nature मतलब जो एक कोडॉन है, this will always code for phenyl alanine. It will never code for any other amino acid. For example, it will never code for leucine, lysine, or it will never code for any other amino acids. Every time, no matter however it is, it will always code for the phenyl alanine. Okay. And next, it is nearly universal. ये थोड़ा confusing हो सकता है कि ये word ना बहुत ही ज्यादा confuse कर सकता है या ये tricky part है, क्योंकि अब अगर हम statement में पढ़ेंगे universal तो दिमाग में रहेगा. So in the exam, if they give the option as like codons are universal, that would be wrong. If they only mention that they are nearly universal, then the statement is right. Why? Because there is some variation. There is some variation in the preference for the different codons. मतलब जैसे कि this codon will always give phenyl alanine. That is fine. But phenyl alanine in every cell, in every organisms, in every species is not always coded by the triple U. Some might prefer only UUC and they have only this codon which is giving the phenyl alanine. ये uniform नहीं है across the species, across the organisms. That is why we say it as nearly universal, जहाँ पे थोड़ा changes है. Next, some exceptions are there. Next, they are commaless. Commaless इसलिए बोलते हैं क्योंकि genetic code जब हम present करेंगे उसमें ना कोई comma होता है ना कोई gap होता है. That is why they are called as non-commaless. And next, there is no non-overlapping, जैसा मैंने बताया था पिछले वाला साइड में कि बीच में जो mRNA है, like wherever there are mRNA, while giving translating its information into the amino acid language, there will be like no overlapping. Single 3 set of mRNA will give one amino acids, next 3 set will give second amino acid, next 3 set will give next amino acids. Okay. Next, they contain specific start codon and stop codon. ये भी पता होना चाहिए कि like whenever there is a like translation that is taking place, whether it is prokaryotes or whether it is like eukaryotes, they have a specific start and stop codon. For example, in the prokaryotes, which is a start codon, it is AUG. In the eukaryotes, it is also AUG. But other than this AUG, for the prokaryotes, there are also 2 more. It is GUG and UUG. So they can also act as a start codon, but even if they act as start codon, they will always code for formyl methionine. ये formyl methionine ही दे देंगे. You can remember this. अगर if they are these two, rarely they act as a start codon, but if they are acting as start codon, they will not code for their respective amino acids. But at this point of time, whenever they are acting as start codon, they will always code for formyl methionine. This is in the prokaryotes. And in the eukaryotes and also in the archaea, ये भी याद रखना, ये बहुत important question है, बहुत ही common question है जो exam में पूछा जाता है. In the archaea and the eukarya, eukarya and the archaea, which is start codon, it is AUG, and it is always codes for only methionine. ये methionine को ही कोड करेगा. It is not formulated. Prokaryotes में formulation हो जाता है. That is why we call it as like formal methionine. But eukaryotes में it is like every time it is a methionine only. It is in both eukaryotes and it is in the archaea. That's what you have to remember. ठीक है. Next, ये तो start codon हो गया. So these are the start codon. Now I don't think like I have to say it for stop codon. You know whether it is like prokaryotes or whether it is eukaryotes. There is a common here. Eukaryotes में हो गया, prokaryotes में हो गया, and archaea में हो गया. Like the stop codons are mainly three, or simply they are three. It is like UAA, UAG, and UGA. And UAA is called as ochre, and UAG it is called as amber, and UGA it is called as opal. ठीक है, three Greek terms है उसके लिए, and they are called as like stop codon, and it is for both prokaryotes, eukaryotes and archaea. ठीक है, and remember the properties of the genetic code are very very important. तो आप लोगों को बिल्कुल बिल्कुल नहीं भूलना है. ठीक है, yes, okay. Next, degeneracy of genetic code का proper example है यहाँ पे. देखो, there is one codon each that codes for methionine and tryptophan. Tryptophan and methionine को कोड करने के लिए सिर्फ और सिर्फ यही कोडॉन्स है. इसके अलावा कोई और कोडॉन नहीं है. There is no other codon which codes for the methionine or tryptophan. They are always coded by one codon. There are some which are coded by two codons. These are the example which are 9 amino acids. इसको as such याद करने का जरूरत नहीं है. Like there is no need of remembering that as such, but just to understand the concept of degeneracy, I have given this table. So don't worry about too much about this slide. एक बार देख लेना, अगर याद करने होंगे तो ग्लांस कर लेना, बाकी ये जरूरत नहीं है. ठीक है, याद करने का जरूरत नहीं है, लेकिन पढ़ लेना. Okay, degeneracy. Next comes like ये थोड़ा पढ़ना चाहिए क्योंकि एग्जाम्स में क्वेश्चंस आ सकता है. Directly without asking like properties, they can ask like they can go one more step ahead and ask you about something about degeneracy. Now, how the degeneracy is resulting into? How more than one codon are coding for same amino acids? There are two possible reasons for this. One is like having more than one tRNA. अगर more than one tRNA with different anticodon, जैसे कि हमें पता है कि this is a structure of tRNA, and यहाँ पे जो तीन bases होंगे, we call it as like anticodon. If there are more than one tRNA with a different anticodon, but all of them, like if I call it tRNA1, there is tRNA2, there is tRNA3, each with a different anticodon. Let's take like this anticodon 1, this is anticodon 2, this is anticodon 3. But all of them, if they are coding for a single codon, वो सारे same amino acid को ही लेके आ रहे हैं. Okay, if that is the case, then that obviously that will result in the degeneracy. So that is a one possible explanations. And the tRNAs, the different tRNAs which can go and bind to the same codon or which can bring the same amino acids, they are called as what? The tRNAs which brings the same amino acids with the different anticodons in them. What do we call it as? It is called as what type? What are they called as? Like what are these tRNAs called as? They are called as like iso-accepting tRNAs. जहाँ पे anticodons different है, but the amino acids that they are bringing, they are same. So these are like iso-accepting tRNA. ठीक है. Next, second possible explanations. यहाँ पे आप देख सकते हो कि see same thing like first point whatever I discussed. See, anticodons are different in the tRNA. Anticodons are different, but the amino acids which it is bringing, it is always lysine. It can code for, it is coding with the different codon, but all the 4 different anticodons, it is bringing the same leucine every time. So all this are called as iso-accepting tRNA. इनको बोलते हैं iso-accepting tRNA. याद रखना, ये एग्जाम में क्वेश्चन आ सकता है. It can come in the exam. ठीक है? Okay. So that is a one possible explanation. Second possible explanation is the very important islet which is called as Wobble hypothesis. Because of the Wobble hypothesis or the Wobble, इसको बोलेंगे Wobble behavior. Not a stringent behavior, that is there is slight variated behavior that we call it as like Wobble behavior. ठीक है? Yes. So the ability of an anticodon to base pair with more than one codon. Same ही anticodon होगा. जैसे कि for example, you take like UUU and UUC. UUU and UCC. So they are having like different anticodon, but they bind or they make a bond with the same codon. so that is what called as Wobble behavior. This is seen in for some of the bases. कुछ bases के लिए हम specifically इसको देखते हैं, and because of this Wobble behavior, even though there are two different anticodons, they are coding with the same codon, because of which like the degeneracy behavior is seen. And this is the Wobble behavior. This is given by Wobble hypothesis. So now you have to understand this Wobble hypothesis for whichever basis we see this Wobble behavior. Wobble behavior कौन से basis पे हम देखते हैं, सारे basis पे नहीं देखते. There are 4 bases, but in not on all the 4 bases we see this effect. We only see in 2 to 3. Okay. Now how to remember this? So I'll tell you that simple trick. एक simple trick बताऊंगा यहाँ पे. See what does the Wobble behavior means? Wobble behavior पहले explain कर दूं क्या है वो. फर्स्ट दो जो पेयरिंग है ना, ये हमेशा स्ट्रिक्ट होता है. इट इज ऑलवेज स्ट्रिक्ट कि जी के साथ हमेशा सी आएगा, ए के साथ हमेशा यू ही आएगा. बट जो थर्ड है ना, थर्ड में ना कभी कभी ऐसा हो सकता है कि ये रैंडमली कुछ भी या कुछ इसमें कुछ स्ट्रिंजेंसी नहीं होता है, कोई अदर बेस भी आ जाए तो चल जाएगा, ये पेयरिंग कर लेगा. सो इफ दैट इज द केस वेयर थर्ड पोजीशन इज नॉट स्ट्रिंजेंट और नॉट स्ट्रिक्ट, जहां पे वोबल बॉन्डिंग हो सकता है, दैट इज व्हाट वी कॉल इट एज Wobble behavior. देखो एग्जाम्पल क्लियर है यहाँ पे. ये नॉर्मल केस है जहां पे जी के साथ हमेशा सी ही आता है. बट इफ इट इज शोइंग द Wobble behavior, जी ना हमेशा सी के साथ ही नहीं, कभी कभी यू के साथ भी पेयर कर सकता है. एंड दिस इज व्हाट वी कॉल इट एज Wobble behavior. Because of this behavior only we see degeneracy in genetic code. And if you have to remember, सारे नाइट्रोजन बेसिस Wobble behavior नहीं दिखाते. So remember, सारे Wobble behavior नहीं दिखाते. There are 3 टोटल, 2 नहीं दिखाते, बाकी 3 है जो स्टैंडर्ड है जो दिखाता है. सो हाउ टू रिमेंबर इट? सो रिमेंबर फर्स्ट वन इज सीए. आई विल टेल यू द सिंपल ट्रिक. यहाँ पे एक सिंपल ट्रिक बता दूंगा. सीए यू नो, चार्टर्ड अकाउंटेंट, दे आर वेरी स्ट्रिक्ट, दे आर वेरी राइट इन द कैलकुलेशंस, दे आर वेरी एक्यूरेट. सो इतना याद कर लेना, सीए मतलब एक्यूरेट है. वो लोग एक्यूरेट है, वो कभी भी मिस नहीं करते, कभी भी कुछ रॉन्ग नहीं करते. सो इफ दैट इज द केस, सी ऑलवेज पेयर्स विद जी विद नो मिस्टेक्स एट ऑल. ये ऑलवेज पेयर्स विद यू विद नो मिस्टेक एट ऑल. कुछ भी मिस्टेक वो बिल्कुल नहीं करते, वो दे आर वेरी परफेक्ट इन देयर वर्क. नेक्स्ट आता है यूजी. हम जैसे लोग यूजी में पागल होते हैं, हम कुछ भी कर लेते हैं, बहुत सारे मिस्टेक्स करते रहते हैं, हम कुछ भी ढंग से कोई काम नहीं करते. सो यू रिमेंबर दिस. सो यूजी, जैसे हम लोग यूजी में थे और तुम लोग यूजी में हो, तो रिमेंबर यहाँ पे हमारा एक्यूरेसी कुछ भी नहीं होता है, हम पागल होंगे, हम गलती करते रहेंगे. सो यू, हाउ टू रिमेंबर इट? यू का स्ट्रिक्ट पेयरिंग कैसा होगा? इफ यू नॉर्मल पेयरिंग किससे होगा? ए से नॉर्मल पेयरिंग होगा. जैसे मैंने लास्ट टाइम ट्रिक बताया था, फंडामेंटल रूल. इफ दिस इज द पिरामिडिन, पिरामिडिन्स ऑलवेज पेयर विद द प्यूरिन. एक प्यूरिन से तो नॉर्मल पेयरिंग हो रहा है, अगर एक प्यूरिन से नॉर्मल पेयरिंग नहीं के अलावा दूसरा पेयरिंग भी पॉसिबल है, देन व्हाट इज द पॉसिबिलिटी ऑफ दैट पेयरिंग विद? इट कैन पेयर विद ओनली द अनदर प्यूरिन. जो एक प्यूरिन के साथ तो नॉर्मल पेयरिंग हो रहा है, बट व्हाट इज अदर प्यूरिन दैट इज पॉसिबल? दैट इज जी. जी इज द अनदर प्यूरिन. सो यू विल either pair with like A or G. Okay, and G is like purine, right? So purine will always base pair with pyrimidine. Normally C से पेयर करेगा, ये pyrimidine हो गया. इसके अलावा हमारे पास क्या बचता है जनरली या नॉर्मली? तो उसके साथ और एक pyrimidine कौन सा बचता है जो स्टैंडर्ड है? वो है C, वो है U. So it will either pair with C or it will either pair with U. Wobble behavior यहाँ पे इस दोनों में देखते हैं हम. Okay, next there is an unusual base which is like inosine. There is some kind of transformation या modification that has been done in the base, that's why inosine. So whenever there is a inosine, it will base pair with either A, U or C. ये pyrimidine भी और purine भी. यहाँ पे याद रखना, G को छोड़ के सारे के साथ ये पेयर कर लेगा इनोसिन. इतना आप याद कर लोगे, सो एग्जाम में डेफिनेटली क्वेश्चन आएगा तो आप आंसर कर सकते हो. सिंपल ट्रिक याद रखना, सीए होंगे बहुत एक्यूरेट होंगे, उन पे बेस पेयरिंग पे कोई प्रॉब्लम नहीं होगा. लेकिन यूजी वाले होंगे जो पागल होंगे हमारे तरह, मैं हूं लेकिन आप नहीं हो, ठीक है, दिल पे मत लेना, बट याद रखना ट्रिक को. सी वी मेक ऑफन मिस्टेक्स करते रहते हैं, तो यहाँ पे मिस्टेक होके एक दूसरे तो पेयर हो जाता है. सो इफ नॉर्मल पेयरिंग नहीं हो रहा है तो अबनॉर्मल पेयरिंग भी पॉसिबल है. ठीक है, यस, गुड शर्ट. अच्छा, थैंक यू सो मच. नेक्स्ट एक्सेप्शन है स्टॉप कोडॉन्स. लाइक वी जनरली नो दैट UAA, UGA एंड UAG. सी ये 3 क्या है? स्टॉप कोडॉन है. बट देयर इज आल्सो एक्सेप्शंस. ये भी यूनिवर्सल नहीं है, इसमें भी एक्सेप्शन है. सी इट हैज बीन सीन इन सम ऑफ द ऑर्गेनिजम्स जहां पे देयर इज 21 अमीनो एसिड्स. स्टैंडर्ड तो 20 है, बट उसके अलावा भी एक्सेप्शन में 2 आ जाते हैं. दैट्स व्हाई लाइक इट इज 22 नाउ. So one is like selenocysteine. Selenocysteine का नाम इसलिए आया क्योंकि cysteine के पास क्या होता है? Cysteine, if you see the structure, it has sulfur. But sulfur के जगह selenium है, तो it is called as selenocysteine. Structure of selenocysteine has selenium in its structure, replacing the sulfur. तो ये selenocysteine है ना, it is coded by UGA, one of the stop codon, जो opal है, and it is been seen in euploids, protozoa इसमें देखा जाता है normally, and it is a rarest case, rare में दिखता है, generally नहीं दिखता, but it has been observed in that particular category. ठीक है. Next, it contains the selenium, बोल दिया, and it is found in small number of proteins, which is involved in redox reaction. जो भी redox reaction में प्रोटीन्स इन्वॉल्व रहेंगे ना, उसके एंजाइम्स फॉर्म करने में, उसके एंजाइम के स्ट्रक्चर में ये वाला सेलोनोसिस्टीन प्रेजेंट होता है. नेक्स्ट, इतना याद कर लेना. नेक्स्ट, देयर इज अ पाइरोलुसिन. पाइरोलुसिन, इट इज कोडेड बाय UAG. दिस इज अनदर सब कोडन व्हिच इज लाइक अंबर है, एंड इट इज बीन सीन इन द आर्किया व्हिच इज लाइक मेथनोजेनिक आर्किया. मेथनोजेनिक आर्किया में इसको कॉमनली देखा जाता है. ठीक है. एंड दिस इज अबाउट द 21st एंड द 22nd अमीनो एसिड्स, जो आप लोगों को पता होना चाहिए. नेक्स्ट, लेट्स स्टार्ट विद द ट्रांसलेशन प्रोसेस. ट्रांसलेशन प्रोसेस को स्टार्ट करेंगे. देखो, जब भी हम ट्रांसलेशन प्रोसेस को स्टार्ट करते हैं, लाइक व्हेनेवर वी वांट टू स्टडी ट्रांसलेशन, वी हैव टू स्टडी लाइक 3 different units, 3 different components. See, 3 different components of translation is like mRNA, rRNA and tRNA. यही 3 important components है. These are the 3 important components because of which translation takes place. तो हम like we have to like separate them and study them clearly, so that in the exam also like this is what they are going to ask. First thing, let's start with the mRNA. mRNA से start करेंगे. So what is the difference between the prokaryotes and eukaryotes? I always said that we will be discussing everything here by just comparing the prokaryotes and eukaryotes. So first of all, what is translation? अगर ये भी बताना होगा, translation is nothing but mRNA. अगर mRNA से अगर amino acids form होंगे, whenever the mRNA is translated, the information that is present in the DNA language, it is translated into amino acid language, that we call it as like translation process, or simply a protein synthesis process. It is a process of synthesis of the protein by decoding or translating the information from mRNA to the amino acid sequence. ठीक है. Okay. mRNA. In case of prokaryotes, there is a single mRNA. एक ही mRNA होगा, but single mRNA will code for more than one polypeptide. ऐसा single mRNA होगा, mRNA will be single, but the polypeptide that comes from this will be like multiple. That is why they are called as like polycystonic. Polycystonic mRNA. Just remember the example of lac operon. Lac operon में क्या होगा कि एक ही promoter है और एक ही operator है, अगर आपको याद हो already, but it has like lac Z, lac Y and lac A gene. देखो, एक ही बार this is mRNA, एक ही बार ये होगा, तो the genes that will come here, it is like lac Y gene आएगा, lac Z का gene भी translate होगा and lac A gene भी translate होगा. Okay, when it is from DNA to mRNA to RNA, sorry, mRNA to amino acids. So that is why like that is what the polycystonic means. But ये भी याद रखना, see, not strictly the prokaryotes are polycystonic. ऐसे भी cases होंगे जहाँ पे prokaryotes भी monocystonic होंगे. What is monocystonic? Just opposite of it. Every promoter and every operator will have a information for single amino acid, single polypeptide chain or single enzyme or single protein. एक ही polypeptide chain या enzyme या protein का information रहेगा. That is what like monocystonic means. That is the strict nature of eukaryotes. Eukaryotes का strict nature है. They are always monocystonic in nature. यहाँ पे polycystonic का cases बिल्कुल नहीं आता. And mRNA in the prokaryotes are very short lived. सिर्फ और सिर्फ 2 मिनट का half life होता है इसका. And if in case of like eukaryotes, it is like quite varying, जहाँ पे 6 hours से 6 hours होगा up to 24 hours वेरी करता है. रेंज इसका बड़ा है. कुछ में तो more than 24 hours like up to 4 days भी देखा जाता है. And in long lived mRNA is also there, like in the lotus, जो genes, some of the genes they usually stays for more than years, like more number of years or several years. ठीक है. Next, when it comes to the ribosome binding site, they have like multiple ribosome binding site. एक ही mRNA होगा, but इसके पास बहुत सारे बाइंडिंग साइट होंगे जहां पे राइबोसोम आके बाइंड हो सकता है. बट व्हेन इट कम्स टू लाइक यूकैरियोट्स, दे हैव लाइक सिंगल राइबोसोमल बाइंडिंग साइट. तो ये भी डिफरेंस याद रखना. नेक्स्ट, सो इनिशिएशन, ये तो हो गया एमआरएनए के बारे में. लेट्स टॉक अबाउट द इनिशिएशन. व्हेनेवर वी टॉक अबाउट द इनिशिएशन इन द प्रोकैरियोट्स एंड यूकैरियोट्स, as I already told you, like AUG is a start codon in the prokaryotes and also it is like AUG in the eukaryotes. But along with AUG, there are other 2 important codons that can also act as start codon in the prokaryotes. It is like UUG and other one is like GUG. GUG है, UUG है, so which can also act as start codon. अगर they are acting as start codon, they will code for formyl methionine only. They will code for formyl methionine, but generally this codes for valine and this codes for leucine. ये लूसीन कोड करता है, वेलिन कोड करता है नॉर्मली, बट अगर ये स्टार्टिंग टाइम पे रहेगा, स्टार्टिंग रीजन में रहेगा जहां पे ट्रांसलेशन स्टार्ट हो रहा है, उस टाइम पे वो फॉर्मल मीथेन को ही कोड करता है, ठीक है। एंड दिस इज समथिंग अनयूजुअल, उसको याद रखना होगा। नेक्स्ट, कमिंग टू द स्ट्रक्चर ऑफ mRNA इन प्रोकैरियोट्स एंड यूकैरियोट्स। यूकैरियोट्स प्रोकैरियोट्स का स्ट्रक्चर में क्या क्या डिफरेंस है, देखो सारे में स्टार्ट कोडॉन है, स्टॉप कोडॉन है, स्टार्ट कोडॉन है, स्टॉप कोडॉन है, एंड इसके बीच में इंट्रासिस्टोनिक रीजन होंगे, ठीक है। दैट इज व्हाई वी कॉल इट एज लाइक पॉलीसिस्टोनिक mRNA इन केस ऑफ प्रोकैरियोट्स। जो स्टार्ट कोडन के पीछे लीडर सीक्वेंस होंगे एंड स्टॉप कोडन के बाद ट्रेलर सीक्वेंस होंगे, दैट कंप्लीट्स द सिंगल स्ट्रेच ऑफ mRNA। एक सिंगल स्ट्रेच ऑफ mRNA में हालांकि मल्टीपल जीन्स के लिए इंफॉर्मेशन हो सकता है, बट दे विल हैव लाइक सिंगल लीडर सीक्वेंस एंड अ सिंगल ट्रेलर सीक्वेंस। दिस इज अ प्रोकैरियोटिक mRNA. If you talk about the eukaryotic mRNA, they are strictly monosystonic. That means they have a coding region for a single gene. Along with this, because of the RNA processing, there will be like 5' capping and there will be like 3' tailing. And adjacent to this 5' capping, there will be like 5' UTR sequence, 3' UTR sequence. What are these UTR sequence? They are nothing but like untranslated regions, जो present होंगे eukaryotes में, 5' में, 3' में. They are similar to that of leader sequence and the trailer sequence. ठीक है? And coming to the binding, कैसे binding start होता है? Remember, in the prokaryotes, it is like 16S rRNA that have a sequence similarity. जो homologous complementary sequences 16S RNA के पास होता है, जो डायरेक्टली आके जो स्टार्ट कोडॉन है, सॉरी जो mRNA है उससे बाइंड हो जाता है, then starts the process of translation in the prokaryotes, and this is how the translation initiation takes place. And here these sequences, whatever you are seeing in the pink region, they are called as what? So if you remember, there are specific sequences which can start the translation process in the prokaryotes. Prokaryotes में एक proper consistent sequence है जहां से ट्रांसलेशन हमेशा स्टार्ट होता है. व्हाट इज इट कॉल्ड एज? यस, वेरी गुड, वेरी गुड. इट इज शाइन डलगार्नो सीक्वेंस. याद रखना स्टूडेंट्स, लाइक दिस इज इंपॉर्टेंट फॉर योर एग्जाम. इट इज कॉल्ड एज शाइन डलगार्नो सीक्वेंस. ये प्रोकैरियोट्स में ही अवेलेबल है, यूकैरियोट्स में नहीं है. एंड यू कैन सी इट इज प्रेजेंट लाइक जस्ट अपस्ट्रीम ऑफ स्टार्ट. देखो ये स्टार्ट कोडॉन है, ये यूजी, इसका जस्ट अपस्ट्रीम में शाइन डलगार्नो सीक्वेंस है. एंड दिस शाइन डलगार्नो सीक्वेंस हैव the sequence complementary with the 16S rRNA. 16S rRNA के साथ complementary है, so that you can go and bind it, and that's how like it starts. And in case of eukaryotes, अगर eukaryotes का बात करें, then instead of Shine-Dalgarno sequence, like not all the eukaryotes, but generally the eukaryotes have one type of sequence. What it is called as? Yes. What it is called as in the eukaryotes? लिखा तो है. Yes, it is called as Kozak sequence. Exactly याद करना. Kozak sequence is nothing but just opposite of Shine-Dalgarno sequence. Shine-Dalgarno sequence prokaryotes में होगा, Kozak sequence eukaryotes में होगा. बट एक चीज है यहाँ पे कि Kozak sequence हर एक स्पीशीज में, हर एक ऑर्गेनिजम्स में नहीं होता है. जनरली होता है, बट हर एक में नहीं होता है. एंड another difference is Shine-Dalgarno sequence are just upstream of transcription start site, but the Kozak sequence are included within the transcription start site. इसके अंदर ही ट्रांसक्रिप्शन स्टार्ट साइट होता है. दैट ट्रांसलेशन, सॉरी नॉट ट्रांसक्रिप्शन, सॉरी इट इज ट्रांसलेशन. ठीक है, ट्रांसलेशन स्टार्ट साइट होता है जहां पे स्टार्ट कोडॉन प्रेजेंट होगा. सी विद इन दैट ओनली लाइक देयर इज अ स्टार्ट कोडॉन इन केस ऑफ यूकैरियोट्स, बट इन द प्रोकैरियोट्स द स्टार्ट कोडॉन इट इज जस्ट adjacent to this of a translation initiation site which is called as Shine-Dalgarno sequence. ठीक है, so these are the two important things that you have to remember for prokaryotes and eukaryotes. Next comes to the next important component which is like tRNA. What is this tRNA? tRNA is nothing but the adapter. For the first time Crick था जिन्होंने, it was by the Crick in 1957 postulated that the correct alignment जो amino acid है उसका correct alignment अगर mRNA से होना है then it needs like some adapter. एक mediator चाहिए जो correct amino acid को लेकर आता है. For example, if starting 3 are AUG, there should be some adapter which carries the methionine amino acids and bring it to exact place and binds with it. And he predicted it to be like अब adapter, उसको adapter molecule बोला. इसीलिए you can also find that tRNA is called as like adapter molecule. Next, later it was known that it is a tRNA which function as the adapter, and this existence of the tRNA for the first time was demonstrated in the same year by Hoagland and co-workers. ये याद रखना name, by Hoagland and co-workers. They were the first one to just identify the presence of the tRNA in 1957. And next यहाँ पे जैसे मैंने पहले बोला था, like I told something in the starting to remember that slide, जहां पे जेनेटिक कोड का दो इंपॉर्टेंट साइंटिस्ट को हमने याद करने के लिए बोला था. वन वाज नीरेनबर्ग एंड सेकंड वन वाज second one was Argobin Khurana. ठीक है? Yes. So after that there is a Robert Hole and the co-workers. They were the first one to determine the sequence of ईस्ट अलानाइन tRNA. जो पहला tRNA को जिसका पूरा सीक्वेंस को पता चला, सीक्वेंस को उन्होंने डिकोड किया, इट वाज फॉर अलानाइन tRNA, अलानाइन कैरिंग tRNA. ईस्ट में अलानाइन कैरिंग tRNA को उन्होंने पूरा डिकोड किया था. नाउ इट वाज बाय रॉबर्ट होले. नाउ व्हाट इज द कनेक्शन बिटवीन रॉबर्ट होले, नायरनबर्ग एंड अर्गोबिन खुराना? कोई बता सकता है? So can anyone tell me? Narenberg. There is some connection between these three. Narenberg, Hargobind Khurana, and Robert Hole. Yes, think and tell me what is the connection between these three scientists? क्या होगा? एग्जाम के लिए इंपॉर्टेंट हो सकता है. 3D मॉडल, होल इन विट्रो सिंथेसिस ऑफ आरएनए, क्लोवर शेप्ड. Yes, 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 yes. Love from Davangere. Okay. Yes, exactly. Kushi, very good. Kushi, what's your good name? Kushi है, कुछ और है? Yes, Allama Prabhu. Yes, very good. Nobel Prize. Dhruv. Yes. याद रखना कि इन दोनों ने प्रॉपरली जेनेटिक कोड को डिसअपियर किया था, डिकोड किया था. ठीक है, वो प्रोनाउंसिएशन छोड़ दो. उन्होंने उस पूरा जेनेटिक कोड को पूरा जो अमीनो एसिड लैंग्वेज है उसका हर एक कोडन का मीनिंग को प्रॉपरली उन्होंने डिकोड किया था. सो दैट वाज द कंट्रीब्यूशन ऑफ दीस टू साइंटिस्ट. अलोंग विद दैट इट वाज बाय रॉबर्ट होले. फॉर द फर्स्ट टाइम he decoded the entire sequence of the first tRNA that was characterized. It was like alanine carrying tRNA. So because of their three contribution, they were awarded Nobel Prize in the Physiology and Medicine. ये याद रखना, ठीक है? इस contribution के लिए इन तीनों लोगों को साथ में Nobel Prize मिला था. So that's what you have to remember. ठीक है? Yes, it's Kushi only. Good, good, good answer. Yes. So this is about the tRNA. What you have to remember. Next, let's talk about the arms. Before that, यहाँ पे ही बता दो. So there are how many? Like it is called as like what is this structure? This is a secondary structure of tRNA. इसको हम secondary structure बोलते हैं and it is called as clover leaf structure. ये clover leaf structure है which is secondary. Primary structure क्या होगा? What is the primary structure of tRNA? It is like just the sequence of nitrogen bases. Sequence of nitrogen bases is a primary structure. Clover leaf is a secondary structure, and there is also a tertiary structure which is like L-shaped structure. L shape is called as like tertiary structure. There are three different structural variants of the tRNA. Yes. Next. So this is a clover leaf model, and which has a how many stems? There are 3 loops and 4 stems. You can see 1 stem, 2nd stem, 3 stem, 4 stem, and there are 3 loops. Loop and stem system here. Now, each of these loops have different like specific function. वो आपके important के लिए exam, तो आपके exam के लिए important है. ठीक है? So each and every loop has some function that is important for your exam. So first thing, if they ask you what about the function of D-arm? D-arm या dihydrouridine का function क्या है? So what you will tell? Whether it is like loop or stem. इसको देखो D arm भी बोल सकते हैं, D का stem भी बोल सकते हैं, arm भी बोल सकते हैं, D loop भी बोल सकते हैं. It can be called as D arm or D loop. So what is the function of it? उसका function क्या है? Anyone? Same. Remember and tell me like what is the function of anticodon? What is the function of like thymine loop? And what is the function of the amino acid acceptor site? कुछ बताया? Amino acid synthetase. Yes. Yes. Very good. Very good. It is like amino acid synthetase. So what is the function of this enzyme? So what is the function of the amino acid synthetase enzyme? Amino acid synthetase, yes, yes, yes. What is the function of it? Batao koi? Yes, remember this 3 different arms, like I'll tell you the functions of each one of them. So first one, starting with the D arm or D loop. So what is the function of this D loop? So it is like charging of tRNA. Yaad rakhna. The key word is like charging of tRNA. Very good. So charging of tRNA. ये tRNA charging of tRNA मतलब what? Like it means like adding of amino acids. जो इसके टॉप है, इसके टॉप में अगर कोई भी अमीनो एसिड आके बैठ जाता है, so that is what like charging of tRNA means. So it is mediated by the D arm or the D loop with the help of this enzyme synthetase. ठीक है, amino acyl synthetase. Amino acid बोला था ना, amino acyl synthetase. Okay, so this is because of this region there is a charging of tRNA that takes place. That is what is important for you. Next one is like anticodon arm. Anticodon arm is this one. This is the anticodon arm. So what is the important functions of anticodon arm? So this anticodon arm has a three important functions. If you remember, if you can tell me, like there are three important functions of anticodon arm. First thing is about codon recognition. ये codon को recognize करेगा जाके properly. Second one is like it gives the Wobble effect. It is responsible for, it is the same arm which is responsible for the Wobble effect. Third one, it is for ribosome binding. Ribosome binding. तीन चीजें हैं, 1, 2 and 3. तीनों याद रखना, एक तो कोडॉन बाइंडिंग के लिए चाहिए, दूसरा Wobble effect के लिए भी चाहिए, and third one it is also needed for the proper ribosome binding. So this is the main function of the anticodon arm. So ऐसे कीवर्ड्स याद करोगे तभी एग्जाम में फायदा है, ठीक है? Like you have to remember the keywords. D arm बोलते ही चार्जिंग ऑफ tRNA याद करो, उसके बाद आपको खुद याद आ जाएगा. Like you will always remember that it is by amino acid synthetase, which is the main enzyme required for the charging. ठीक है? Yes. Next one. Next one is like thymine arm. The third one is like thymine arm. So what is the important function of the thymine arm? Thymine arm या thymine loop. It is an unusual nitrogen base which is not seen in which generally is not seen in RNA, but it is found in this tRNA, and it is with the another base which is like pseudouridine. Thymine के साथ और एक unusual base होगा यहाँ पे. It is in the thymine loop or thymine arm. It is pseudouridine. So what is the main function of this pseudouridine or this entire arm or loop? Thymine का क्या function होगा? Yes, yes. Anyone? So this is a important region which functions as a ribosome attachment. जो ribosome main attachment के लिए जो दो subunits आके attach होंगे, so उस जो राइबोसोम का कॉम्प्लेक्स में जाके प्रॉपर्ली टीआरएनए को अटैच होने के लिए, दिस इज द मेन आर्म व्हिच इज रिस्पांसिबल फॉर इट, राइबोसोम अटैचमेंट. Remember the keyword, the ribosome attachment. Next, there is one more arm which is called, we'll go to the variable arm later, but let's complete this amino acid acceptor arm. Amino acid acceptor arm is the top one here. This is called as like amino acid acceptor arm. So what is the main function of amino acid acceptor arm? As per the name, नाम ही बोल रहा है उसका. So it is the one which is like responsible for the attachment of specific amino acid based on the anticodon. Anticodon के basis पे specific amino acid को attach करने वाला enzyme. This is like amino acid acceptor arm. Okay, so these are the four different like arms or loops that is important. Along with it, four arms and 3 loops, there are one more arm which is called as variable arm. First of all, so why the name variable arm? Variable arm is named because the length of this arm, the extra arm, is like variable. इसका जो length है ना, ये uniform नहीं है across the species, across the organisms. It is like variable. That is why the term variable arm. Okay. Exact functions तो पता नहीं है, लेकिन it is mean to be like playing an important role in tRNA recognition by the enzymes which are attaching the amino acids. ठीक है? Amino acid synthetase जो चार्ज करता है उसमें भी थोड़ा इसका भी contribution है. That is what it is predicted to be. But exact function इसका पूरा clear नहीं है. And it also facilitates the folding of tRNA, जो 3-dimensional structure बनता है L shape में, and it is also predicted that it also plays an important role in the L shape formation or L shaped structure formation. Okay, this is about the arms and the loops of the tRNA जो exam में directly exam में आ सकता है, like that can come in the exam. So please, if you are not very sure about it, अगर आपको पहले पढ़ा है तो याद नहीं है तो at least अब से याद रखना. If I have to recall, D arm, it is charging of tRNA. Amino acid acceptor arm, it is accepting the amino acids, correct amino acids based on the anticodon. Next, if it is like pseudouridine or thymine loop, it is about the ribosome attachment region. And if you talk about the anticodon arm, it is for the 3 important functions. One is for codon attachment, proper codon attachment. Second one is like Wobble effect. Third one is like ribosome attachment. ठीक है? Yes. Which dihydrouridine, yes, dihydrouridine, DHU, yes, it is also called as DHU loop. ठीक है? D loop या DHU loop, dihydrouridine. Yes, anticodon arm also ribosome attachment. Yes, like it plays a significant role in attaching the ribosomes. Those proper site provide करता है, that is with the help of this arm. Okay. Yes, so this is what you have to remember. Next comes the third important component of this translation, which is the rRNA. rRNA, that is another very important जो exam में directly exams में questions आ सकते हैं. So next, so rRNA which is the most abundant in the cell, जितने भी RNAs है उसमें rRNA ही है, ribosomal RNA. The other name of the full form of this rRNA is like ribosomal RNA, which is like most abundant, and the order of its abundance starts with like rRNA, tRNA, and last it is mRNA. mRNA सबसे कम होता है. Next, the rRNAs are important. Why they are important? Because they are important component of ribosomes. Ribosomes का important component है. Or like ribosomes, if you have to decode, ribosomes are made up of two important components, which is like one is rRNA and second one is protein. एक ribosomal proteins होंगे, एक ribosomal RNA होगा. Okay, so this is what the ribosomes are made up of. And what are ribosomes? They are small migrating factory which physically moves along the mRNA. जो mRNA होगा उसके ऊपर दो subunits आके attach होंगे, but they physically move along with the structure of mRNA, and that's how like synthesis of translation, or that's how the synthesis of protein takes place. And in the comparing the prokaryotic ribosome and the eukaryotic ribosomes, दोनों में क्या difference है? देखो, first one is like 70S and 80S. We all know it already. ये 70S बोलते हैं, and this we call it as like 80S. And in the low magnesium concentration, it splits up into two different units, one smaller unit and one bigger unit. Smaller unit in the prokaryotes is 30S and bigger unit is 50S. And in the eukaryotes, it is like 40S smaller unit and bigger unit is 60S. 60-40 होता है, 50-30 होता है. And the smaller unit, the component of smaller unit, rRNA, 16S rRNA, याद रखना, 16S rRNA is present in the smaller subunit with the 21 proteins. प्रोटीन का नंबर्स यहाँ पे भी बोल दो कि प्रोटीन का नंबर्स बुक टू बुक में वेरी कर रहा है, सो डोंट वरी अबाउट द प्रोटीन. सो आइदर यू रिमेंबर whatever it there in the board or whatever you are read it from the book. दोनों याद रख लेना, दोनों दे के कंफ्यूज बिल्कुल नहीं करेंगे एग्जाम में, so don't worry about it. But otherwise like don't think too much or don't go dig deeper into this protein. So it is variable in the different books. ठीक है? Yes. And the smaller unit is like 16S rRNA in prokaryotes, smaller subunit has like 18S rRNA in the eukaryotes. And when you talk about the larger subunit, it has like 23S rRNA, and in the eukaryotes it is like 20S rRNA. 23 है, 28 है. उसके अलावा 5S rRNA होता है, and there is also 5S rRNA in case of eukaryotes. But along with the 5S, ये भी याद रखना, 5S के साथ 5.8S. एक 5.8S rRNA होता है, that is specific to the eukaryotes. अगर एग्जाम में आपको ये यूनिट्स के बारे में पूछा जाएगा, like you have to answer it. In the prokaryotes, it is like 16S rRNA in case of smaller subunit, and it is like 23S rRNA in case of 18S rRNA in case of eukaryotes. And if you talk about larger subunits, it is like 23S rRNA, and eukaryotes it is 28S rRNA. But apart from that, there is also one more which is like 5.S rRNA, that is specific to the eukaryotes, which is not found in the prokaryotes. इतना सबको पता होना चाहिए. Okay, like this is what you have to remember. Number of nucleotide important or not in the components of ribosomes. Number of nucleotides, इतना deep में मत जाओ, इतना important नहीं है. But remember like about the subunits of both larger subunit and the smaller components of larger subunit and smaller subunit in both prokaryotes and eukaryotes. It is like 16S rRNA, 23S rRNA in case of prokaryotes, plus 5S rRNA, which is common to both prokaryotes and eukaryotes. But when you talk about the eukaryotes, it is 18S rRNA in case of smaller subunit and 20S rRNA in case of larger, plus in addition to that there is also 5.S rRNA. इतना याद रखना. This is what you have to remember. Next, talking about the structure of the entire ribosome. In the structure of entire ribosome, there are three important sites, which is called as like E, P, and A site. So E is like exit site, you can remember it. P site, it is a peptidyl tRNA site, and A site, it is like amino acid tRNA sites. So first thing, so what is the function of like A site? A site welcome करता है incoming tRNA which is charged with the amino acid. जो भी tRNA चार्ज होता है अमीनो एसिड के साथ, उस tRNA को कौन वेलकम करता है? इट इज वेलकम बाय द ए साइट. ठीक है. नेक्स्ट, व्हाट इज द फंक्शन ऑफ द पी साइट? मैं बहुत ही कैजुअल लैंग्वेज में सिंपल वर्ड्स में बता रहा हूं ताकि आपको याद रहे. ए साइट, इट विल वेलकम द इनकमिंग tRNA which is charged with the amino acids. The P sites which will build the peptide bond and connect all the amino acids, and there is a E site where the deacylated, जहां पे चार्ज को रिमूव कर दिया गया, जहां पे अमीनो एसिड को रिमूव कर दिया जाता है, वो वाले tRNA that comes to the E sites and that leaves the complex. वो E site आके ही complex को लीव करता है. Okay, so that is about the three important sites of the ribosomes. Next, So activation कैसा होता है, like how does this tRNA gets attached to the amino acids? Amino acids को क्या बोलते हैं, like we call it as like charging of tRNA. See, attachment of like amino acids to the tRNA, we call it as like charging of tRNA. वो कैसा होता है? It is by the two-step process. ये two-step process है. It is two-step amino acylation reaction, which is mediated by the amino acyl synthesis. एंड जिसमें फर्स्ट स्टेप में एटीपी एक्टिवेट करता है अमीनो एसिड को, एंड सेकंड स्टेप में अमीनो एसिड इज ट्रांसफर टू 3, जो टीआरएनए का सीसीए साइट होता है वहां पे आके अमीनो एसिड अटैच हो जाता है. दैट इज अ टू स्टेप प्रोसेस ऑन हाउ द अमीनो एसिड इज चार्ज्ड विद द टीआरएनए और टीआरएनए इज चार्ज्ड विद द अमीनो एसिड्स. ठीक है, यस. नेक्स्ट, so translation, next process, let's talk about directly. देखो हमने we discussed in detail about the 3 important components. One is like mRNA, second one is tRNA and rRNA. पूरा डिटेल में हमने तीनों components के बारे में डिटेल में पढ़ा था. Now the point starts with the initiation. How the translation initiation takes place in the prokaryotes and how does it takes place in the eukaryotes? See, in the translation of the prokaryotes initiation, it begins with the smaller subunit से start होता है, and because of the binding of the which rRNA, 16S rRNA. 16S rRNA, since it has like sequence similarity or complementarity with the chain Delgorno sequence, that goes and binds first. Because of that binding, the entire complex binds, and then we have the start codon, which is like main start codon in the prokaryotes. But other than that, we also have like 2 other which can act as a start codon, through which like the translation process begins in the prokaryotes. Okay. Next, when you talk about the eukaryotes along with this start codon, they have like specific initiation factors which are like EIF2, eukaryotic IF2, initiation factor 2, initiation factor 1, 3, 5. इसको as it is आप लोग अगर याद करोगे, it's well and good. ऑप्शनल है, मैं ऑप्शनल इसलिए बोल रहा हूँ कि इसको अभी तक एग्जाम में पूछा नहीं गया है, बट if it is not asked, possibilities are also there, it can be asked or it can be again skipped. अगर याद कर पा रहे हो, like if you are able to remember the function of each one of this, do it. अगर आपका बेसिक क्लियर है, आप सब कुछ क्लियर कर लिए हो, आप सिर्फ रिवाइज ही कर रहे हो, तो ये भी याद रख लेना. अगर आप बेसिक्स पे ही अभी तक टिके हुए हो और वहां पे भी याद करने का मुश्किल हो रहा है, तो यू कैन स्किप दिस पार्ट, इट इज ऑप्शनल. ठीक है, सो दिस इज अबाउट द इनिशिएशन, ट्रांसलेशन इनिशिएशन. नेक्स्ट, व्हेन यू टॉक अबाउट एलोंगेशन, initiation याद रखना, whether it is prokaryotes or eukaryotes, it is the smaller subunit which will go and bind to the mRNA. Prokaryotes में it is like 16S rRNA, in the eukaryotes it is like 18S rRNA. ठीक है, and when it comes to the elongation, again it is by the elongation factors. In the prokaryotes there are 3 important elongation factors, which is like TU, TS and G. Thermo-unstable, thermostable and EFG, elongation factors G. What is the function of TU? It mediates the first entry of amino acid tRNA into the A site. A site पे जब just A site है, like whenever the tRNA comes to the A site, it is because or it is mediated by this elongation factor T. Next, what is the function of like TS? It mediates the release. जो यहाँ पे आया है, ये P region तक आएगा when it comes to P region and when it goes to the E site. So during this time, this is the one which helps in the release of this EFTU. Next, EFG, the last one. So it mediates the translocation. जैसे movement होता है ribosomes का, it is mediated by this elongation factor EG. ठीक है. In the eukaryote, similarly, if you compare, see this is EF1 is similar to TU. Eukaryotic elongation factors 1A. It brings the amino acid tRNA to the A site. Next, the EF2. It is similar to that of EFG, which makes the hydrolysis, GTP hydrolysis, and makes the movement of the ribosomes over the mRNA. Okay, so this is the two important factors, elongation factors that you can remember in the eukaryotes, and the 3 important elongation factors in the prokaryotes. Okay, so this is important. अगर पूछेंगे तो इससे ज़्यादा कुछ भी नहीं पूछ सकते. Maximum अगर कुछ पूछ सकते तो they can ask you about the functions, something about this in the statement, or something about these 2 important elongation factors in the eukaryotes. That's it. ठीक है. One more point, the large subunit. The process of elongation. The process of elongation repeats peptidal transferase activity. Peptidal transferase, if anyone can tell me what is the function of the peptidal transferase? Elongation के time पे peptidal transferase का activity आ जाता है. So what is the main function of it? क्या है मेन फंक्शन उसका? Peptide bond formation. Yes, so it is a peptide bond formation and increasing the chain of amino acids. So that is done by this peptidyl transferase, and this peptidyl transferase activity lies in the larger subunit, which is 20S rRNA in case of eukaryotes, and it lies in the same larger subunit in prokaryotes, which is 23S rRNA. ये याद रखना. In case of prokaryotes, it is 23S rRNA where the peptidyl transferase activity resides, which has a role in making the peptide bond formation and elongation of the chain of amino acids. And in the eukaryotes, it resides in the 20S rRNA, which is again the largest subunit in the eukaryotes. Next, the last step of this translation, very simple. Last step पे आ गए हम. Last step is like termination. Termination कैसा होता है? Prokaryotes में, eukaryotes में क्या common है? Common वही है कि they are the same 3 stop codons which are there, which will stop the translation process. In the prokaryotes, again, देखो initiation time में भी initiation factors थे, elongation time में भी elongation factors थे, and even in the termination there are release factors. Here it is called as like release factors. In the prokaryotes, it is called as RF1, RF2, and RF3. RF1 recognizes and stops the 2 important stop codons, which is like UAA and UAG. And the RF2, it stops the another 2, which is like UAA, which is common to both RF1 and RF2, and the other one, which is like UGA, which is coded for Opal. So RF1, it recognizes the 2 stop codons. RF factor 2 in the prokaryotes also recognizes 2 stop codons. The common between these two are like UAA. Okay, ठीक है. And next, what is the function of RF3? So RF3 stimulates the release of RF1 and 2 जो attach होते हैं stop codons पे, उसको release करने में जो help करता है, that is like RF2. ठीक है, with the help of like GTP energy use करके GTP hydrolysis, just like ATP hydrolysis, it is a usage of energy. Because of that only the entire complex get disassociated, and that's how like the termination of the translation takes place. Similarly, in case of eukaryotes, same ही है, UAA, UAG, UGA, but difference क्या है? In eukaryotes, there is a single, the RF factor 1, eukaryotic RF factor 1. So that is the universal release के लिए काम करता है for all 3 codons. जो all 3 stop codon को stop करने वाला कौन है? It is like eukaryotic RF factor 1. ठीक है? Yes. So and next RF factor 2. Eukaryotic RF factor 2 इसका काम क्या है? What is a function of this? So this has the same function of RF3. RF3 जो है इसका similar function है where it is like involved in the GTP hydrolysis and helps the RF factor 1 to recognize the stop codons. इस stop codons को recognize करने में help करता है. And the RF factor 3 in the eukaryotes, it is also another GTP binding protein that recycle the RF1 and RF2. तो RF3 and sorry, यहाँ पे ऐसा याद रख लो, eukaryotic RF3 it is similar to the function of prokaryotic RF3, जहाँ पे जिनका काम क्या होगा, RF1 and RF2 को properly monitor करना और उसका काम को देखना. सेम चीजें prokaryotic में भी होता है और eukaryotic में भी होता है. डिफरेंस बहुत ही सिंपल है कि in the eukaryotes there is a single release factor that recognize all the 3 stop codons. In the prokaryotes, RF1 recognizes two stop codons and RF factor 2 recognizes 2 stop codons. The common between these two are like UAA is the common between them. So इतना याद रख लेना. देखो ट्रांसलेशन हो गया, ट्रांसक्रिप्शन हो गया या रिप्लिकेशन हो गया, देयर इज अ फंडामेंटल रूल. एक फंडामेंटल रूल है, इट इज लाइक 5 प्राइम टू 3 प्राइम डायरेक्शन. इसमें कोई कंफ्यूजन होने वाले बात ही नहीं है. ठीक है, देयर शुड बी लाइक नो कंफ्यूजन हियर. कुछ भी कंफ्यूजन होना नहीं चाहिए. ठीक है, इट इज द डायरेक्शन इज लाइक यूनिवर्सल. इट इज लाइक 5 प्राइम टू 3 प्राइम डायरेक्शन. ये याद रखना. ठीक है, यस. एंड if you talk about the another fundamental rule, whether you talk about the replication, talk about the transcription, or you talk about the translation, so what are the different steps? इनका steps क्या है? See, all three have the same steps like initiation, elongation and termination. वही 3 steps है जो तीनों में common है. It is like initiation, elongation and termination. ये common है, ये 2 चीजें common है. So remember, these are the 2 fundamental things. Next, there is one more thing called as like polyribosome, or normally they are called as polysomes. Polysomes are nothing but they are like complex of multiple ribosomes which simultaneously translate. एक mRNA आ गया सिर्फ एक ही राइबोसोम जाके अटैच होके एक बार प्रोटीन नहीं करेगा, उसके बाद दूसरा नहीं, multiple ribosome complex attack the same mRNA and they will simultaneously translate the information or give the amino acid. जैसे कि आप देख पा रहे हो single amino acid है but there are multiple ribosomes that have simultaneously attached to it and they are performing the translation process and they are giving the amino acids in n number. Multiple amino acid sequence same mRNA का at the same time produce हो रहा है, that is because of the presence of this polysomes or the polyribosomes. ठीक है? Okay. Now comes the next part is about knowing the clear cut difference. एक comprehensive table है जो clearly define करता है, differentiate करता है prokaryotes and eukaryotes translation process को, the entire translation process. एक दो step नहीं, पूरा translation process का मैंने difference दे रखा है, तो class के बाद इसको एक बार पढ़ लेना. दो slides है but only दो slides है, तो इसको एक बार पढ़ लोगे तो जितना भी हमने discuss किया था, like whatever we have discussed till now, everything will be very clear to everyone. एक बार करना होगा क्योंकि एग्जाम के लिए बहुत इंपॉर्टेंट. सर आप तो लाइक यू आर सेइंग लाइक एवरीथिंग इज इंपॉर्टेंट, स्किप क्या करना है? सी व्हाटएवर वाज नीडेड टू बी स्किप्ड दैट आई ऑलरेडी स्किप्ड इट, उसको मैंने स्किप करके ही ले आया हूँ. सो व्हाटएवर आई एम सेइंग हियर दे आर ऑल इंपॉर्टेंट. ठीक है, इसको कुछ स्किप करना नहीं है. इतना ही सिंपल को स्किप करोगे तो क्यों लोगे रैंक चाहिए क्यों है? ठीक है, सो डोंट डू इट. सर रिमेंबर एवरीथिंग. नेक्स्ट इज अबाउट रेगुलेशन. ये थोड़ा देखने में लाइक डर लगता है कि रेगुलेशन क्या है. लाइक इफ यू रीड फ्रॉम द बीडी सिंह लाइक यू विल हार्डली अंडरस्टैंड लाइक 10 to 20% ऑफ इट. हाँ, लाइक यू कैन अंडरस्टैंड द लैक ऑफ ओरॉन, ट्रिप्टोफैन ऑफ ओरॉन, बट अदर देन दैट द थिंग्स आर नॉट वेरी क्लियर. बहुत ही क्लियर नहीं है. लेट्स ट्राई टू मेक इट सिंपल. थोड़ा सिंपलीफाई करके पढ़ेंगे इसको. रेगुलेशन पार्ट, रेगुलेशन ऑफ प्रोकैरियोट्स एंड यूकैरियोट्स. So what is regulation? Why it is required? See, if you remember, like if you understand this logic, so cell में बहुत सारे genes होंगे, एक organism में बहुत सारे genes होंगे. For example, in the human, there are 25,000 plus genes that are present. So do you think like from the birth to death, all genes are secreted all the time? All genes are secreted all the time or translated and expressed all the time? No, right? So there are specific age related characters or traits that are seen. For example, mustache and beard. So when it comes only after adolescent age, so there are certain other organs like facial hairs, you say like body hairs that comes after some age, certain age, right? So similarly, like so there are multiple characters which are like age specific. कुछ age specific होंगे, कुछ tissue specific भी होंगे. इसी tissue में कुछ expression हो रहा होगा, इस tissue में कुछ specific expression हो रहा है, तो the expression even though the entire body is made up of like same genome, इसमें जितने भी multiple cells है सारे में सेम ही जीनोम है, बट डिपेंडिंग ऑन वेयर दे आर लोकेटेड, बेस्ड ऑन दैट द एक्सप्रेशन चेंजेस. सिंस वी वांट सच टाइप ऑफ मैकेनिज्म वेयर all genes are not to be expressed all the time, we need the regulation. Otherwise, like energy will be wasted. There will be like wastage of energy. That's why the regulation of the gene expression takes place, because all genes are not required to all the times to express, and they are only required to express in the specific life cycle. And in contrast to that, there are certain genes, कुछ genes तो ऐसा है जो regulation होना है, but there are also some genes जो बर्थ से लेकर डेथ तक एक्सप्रेस होते ही रहते हैं सारे सेल्स में. दे आर कॉल्ड एज लाइक हाउसकीपिंग जीन्स व्हिच आर एक्सप्रेस ऑल द टाइम, जो ऑल द टाइम एक्सप्रेस होते हैं. ठीक है, नेक्स्ट द डिफरेंस बिटवीन द प्रोकैरियोट्स एंड यूकैरियोट्स. रेगुलेशन में प्रोकैरियोट्स और यूकैरियोट्स में क्या डिफरेंस है? सी व्हेन यू टॉक अबाउट द प्रोकैरियोट्स, आई टोल्ड यू लाइक प्रोकैरियोट्स हैव लाइक कोड ट्रांसक्रिप्शनल ट्रांसलेशन. जैसे ही एमआरएनए बाहर आया, ट्रांसलेट होकर इंफॉर्मेशन डिसमिनेट हो गया. जैसे ही mRNA आ गया, ribosome will attach and there will be simultaneous translation along with the transcription that will take place in prokaryotes. There is no as such time gap because there is no RNA processing that is needed in case of mRNA of prokaryotes. So that's how it is. No splicing is required, but normally splicing is not seen in the prokaryotes, but it is a strict rule. There are also like some self-splicing introns that are present in the prokaryotes too. ठीक है? Yes, housekeeping genes are also called as like constitutive genes which are expressed all the time. Okay, so that's why we say that as जो major regulated, major control कब कर सकते हो? अगर हम किसी जीन को कंट्रोल करना है कि वो एक्सप्रेस होना है या नहीं होना है, if you want to change something or do something modification, it is only possible in the transcription level in case of prokaryotes. but क्योंकि एक बार ट्रांसक्रिप्शन हो गया तो रुकता ही नहीं है, simultaneously translate होके इन्फॉर्म प्रोटीन आ ही जाता है. So that's why the major step here is in the prokaryotes in the transcription. But यूकैरियोट्स में बहुत ही कॉम्प्लेक्स है, हर एक स्टेप के बाद हमें थोड़ा ब्रेक मिल जाता है. फॉर एग्जांपल देयर आर डिफरेंट लेवल्स ऑफ रेगुलेशंस. ट्रांसक्रिप्शन हो गया, ट्रांसक्रिप्शन के बाद लाइक प्रोसेसिंग होगा, प्रोसेसिंग के बाद एक्सपोर्ट होगा, एक्सपोर्ट के बाद राइबोसोम में जाकर अटैच होंगे, सेम थिंग. बट देयर आर लाइक time difference है, time gap है यहाँ पे. So there are multiple levels of regulation that is possible, which are normally divided into 5 to 6 levels. And no introns removal generally, and there is a removal of introns, and there is no 5 prime capping or tailing that is seen in the prokaryotes, but in the eukaryotes we see this, and this is a part of RNA processing. Okay, this is the main difference between the prokaryotes and eukaryotes. And levels of regulations. The levels of regulations can be divided into two different types. One is like long-term regulation and the short-term regulation. So short-term regulation is just for the immediate need of a cell. It is mostly seen in the prokaryotes. And long-term regulation, it is for the growth and development. It is seen in both prokaryotes and eukaryotes. दोनों में देखा जा सकता है. But short-term regulation जो है mainly वो prokaryotes में देखते हैं. Next, what is the other differences between the prokaryotes and eukaryotes? So I told you, like, this is most common and prominent during the transcription. When you talk about the eukaryotes, first level at a transcription level, second at a post-transcription level, third regarding during the transport of mRNA, fourth during the destroying or keeping it, next during the translation level, and lastly there is post-translation level. Levels multiple books पे multiple levels दिए होंगे, but अगर if you want to simplify it, you can remember like this. पहले transcription में हो सकता है, उसके बाद processing में हो सकता है, उसके बाद transport पे mRNA transport के time में regulation हो सकता है, and it might be during like whether to keep it or not, whether the mRNA can be degraded or not, उस stage में भी हो सकता है. Next, it also can be during the translation level, and even after translation also, even after like polypeptides have been formed, there is a possibility that they can be regulated, yeah, they can be modified so that the protein function can be altered. Yes, everyone. Right. Luxury genes are something which are only expressed in a specific tissue, specific part of a cell. Sorry, specific part of a body. कुछ specific cells होंगे, उसी cells में अगर वो express कर रहे, throughout the body express नहीं कर रहे, so that is what luxury genes means. ठीक है? Yes. Next, understanding the regulation. Regulation को understand करने के लिए हमारे पास दो चीजें. First of all, see, logically tell me like what body में cells में ऐसा कौन सी चीजें हैं जो regulate कर सकता है? Like what are the things that are present in the body which can regulate the gene expression? Control करने के लिए हमारे पास क्या है? Yes, self-splicing introns are the one which are present even in the prokaryotes that do not need like spliceosome complex. Splicing के लिए एक spliceosome complex चाहिए होता है, जो मैंने last class में बताया था, SNRPs, right, which are made up of snRNA plus the protein. So without the spliceosome complex, if the introns are able to splice themselves, they are called as like self-splicing introns. They can be also present in the prokaryotes. Okay? Yes. Now enzymes are the protein. Very good. So proteins are the one. देखो proteins ही है जो regulatory role कर सकते हैं. Whenever I say there are regulatory factors, you have to understand that I am literally meaning to, or I am just addressing the proteins. अगर हम कोई भी regulatory factors करके नाम लेते हैं ना, तो याद रखना we are relating it to the proteins only. So now based on the kind of regulation takes place, it can be by the activators or the repressors. Regulation can be by the activators or repressors. So what are these activators and repressors? One common terminology you have to understand is like both are proteins only. Both are proteins or the enzymes. Enzymes ही है जो proteins है which will regulate the gene expression. How about activators? As per the name, if the gene is getting expressed, upregulated, we say like upregulated, it is because of the activators. If there are proteins which are upregulating the gene expressions, they are called as activators. If there are proteins which are down regulating any expression, कोई भी expression को down regulate कर रहा है, those proteins are called as repressors. Very simple words or simple meaning to understand. कोई भी protein ले लो, अगर कोई भी gene expression को up regulate कर रहा है, those are called as activators. अगर कोई भी protein अगर down regulate कर रहा है, repress कर रहा है, they are called as repressors. So this is what the terms means. Okay. Next, so based on this understanding, there was an operon model that was published in, or that was proposed in 1961. It was by two scientists, Jacob and Monod, and you people will tell us like whether they have did or proposed this operon model independently or they did it together. मुझे ये चीज comment section में चाहिए. आप लोग बताओगे कि independently propose किए थे या दोनों मिलके ये propose किए थे. Okay. And what does this operon explains? Operon explains like how the genes are regulated at the transcription level. Transcription level पे operon concept क्या बोलता है? Like what does it explain? It explains that how the genes can be regulated during the transcription level. Transcription जब होता है उस level पे कैसे regulation possible है, that is explained by the Operon model, and one clear example under the Operon model which explains this is the concept of lac operon. That is why it is famous. Okay, and what is operon? Operon is nothing but a stretch of DNA sequence, which is a sequence of adjacent genes which includes the structural genes like lac, Z, Y, A, and with the single promoter and the cis acting regulatory sequences operator under one transcription control. That means if this is the DNA and it has a promoter region, it has an operator region, it has a complete structural region, structural genes region, okay, and this promoter regions also have like regulatory sequences all under single transcriptional control. ये एक ही यूनिट है ट्रांसक्रिप्शन, मतलब एक ही बार में ये ट्रांसक्राइब होंगे एज अ mRNA, then this is called as a operon. इसको ही एक operon बोलते हैं. Okay, operon are commonly seen in prokaryotes but not entirely specific to prokaryotes. ऐसा नहीं है कि सिर्फ और सिर्फ prokaryotes में ही है, and again it is not common in the eukaryotes. Eukaryotes में बहुत ही rare cases में, बहुत ही lower eukaryotes में दिखता है, but it is generally seen in the eukaryotes ऐसा बोल सकते हैं, but we can't strictly say that it is completely absent in the eukaryotes, ऐसा बोल नहीं सकते. ठीक है? Yes. Okay. Next one. So regulation mechanisms with the help of prokaryotes and eukaryotes कैसा होता है? Like how does it takes place? See, regulation with respect to the activators and repressors I told you, अगर if there is absence of both repressor and activators, दोनों नहीं है, gene expression कैसा होगा? There will be like basal level, एक optimum level का transcription होता रहता है. Optimum level of gene expression will be taking place. If there is a repressors which are present, I told you like down regulation, there is a below the basal level of transcription that will take place. If there is a presence of activators with no repressors, then there will be like above the basal level of transcription. ठीक है? So in the second step, also understand in the presence of repressors with or without activators. देखो repressor जब present होंगे ना, उसको मैटर नहीं करता कि उसके पास एक्टिवेटर्स है या नहीं, वो बिल्कुल सप्रेस ही करेंगे. वो हमेशा डोमिनेंट होते हैं, वो सप्रेस कर देंगे. So whether they are present with the activators or without the activators, they will always down regulate. That's why like there will be always the below the basal level of transcription takes place. But whenever the activators are present without, this is a strict condition, without repressor, यहाँ पे repressor नहीं होना चाहिए, only then there will be like above the basal level या upregulation of any gene expression. ये fundamental rule है, इसको याद रख लेना. इसके बेसिस पे कुछ भी क्वेश्चन पूछा जा सकता है, या lac operon भी इसी के अंडरस्टैंडिंग पे ही explained हुआ है. Okay, now what are the functions of activators and repressors? उनका specific nature है, functional में भी specific nature है, like that we have to understand. See, activators, what are the different regions I told you? Let's take an example of lac operon. इसके पास खुद का promoter है, खुद का operator है, and there is structural genes lac, Z, Y and A. Okay, उसके बाद stop codon होगा, sorry, उसके बाद terminator regions होंगे, since we are talking about DNA. ये DNA है. Okay, now so activators का काम क्या है? So activators have the characteristic feature, they will always go and bind to the promoter. Activators के पास promoter region को bind करने के लिए sequences होता है, sequence similarity होता है, so they always go and bind to the promoter. So what happens when the activators bind to the promoter? Whenever they bind to the promoter, they will increase the binding ability of the promoter with the RNA polymerase. जो एक main enzyme है, जो the main enzyme which is responsible for the transcription of any gene, so that RNA polymerase can come and easily bind to the promoter region if the activators are already binded to the promoters. They will make a conformational changes in the structure of promoter which can easily recognize the ribosomal RNA polymerase, and because of which there is a upregulation of gene expression. जहाँ पे quickly RNA polymerase जाके जल्दी bind हो सकता है, and there will be like transcription that takes place. This is a function of what? This is a function of activators. याद रखना, activators. Next, similarly, what is the other one we have? Like we have repressors. What is the main function of repressor? They have a binding site at the operator region. Repressor का binding site कहाँ है? They have the binding site at the operator region, जो आके clearly bind हो जाता है. But what happens like binding of operator region के पास bind होने से क्या होगा repressor? See, if the operator region is binded with the repressor, the RNA polymerase where the binding site is in the promoter, it cannot bypass it. So because of which the transcription will not take place, or there will be like below the transcription, below the basal level of transcription. Okay. Yes. So this is what you have to understand about the activators and the repressor. I hope it is clear. Next. So based on this behavior, there are 4 different possibilities of the regulation in the prokaryotes. Sorry, eukaryotes. Sorry, prokaryotes. So बहुत ही simple way में अगर समझाना चाहूँगा, like this is a very simple way of understanding the regulation. इसको अगर आप समझ गए ना, तो it's quite easy for you to understand all other things. See, there are 4 different possibilities. First is like for positive inducible, negative is like negative inducible, third one is positive repressible, and last one is like negative repressible. First thing, the example for negative inducible system is like lac operon, which is mediated by lac repressor. तो यहाँ पे positive and negative का बारे में बोलना ही पड़ेगा. So how the positive and negative term is derived? Okay. Now this term is derived based on whether the regulation is by repressors or activators. Whenever there is a gene regulation that is taking place with the help of repressors होंगे जो mainly contribute कर रहा होगा regulation पे कोई भी gene का, then it is under the negative control. क्योंकि repressor का काम ही क्या है? It will repress the operon. That's why it is called as negative system. If any gene regulation, it is because of the activators. Obviously, like presence will enhance the gene expression. Its absence will not enhance it. So that's why they are called as like positive. Repressors उल्टा है. जब वो present रहेंगे तो transcription off रहेगा. जब वो absent रहेंगे transcription on रहेगा. That is why it is called as negative. So I hope like everyone understood why the term positive and negative. Same. Next, apart from this positive and negative, there are 2 other terms. One is like inducible system and next one is like repressible system. So what is the difference between the inducible and repressible system? See, inducible and repressible is derived because of some product, some initial product that will help in the transcription. So here the lac operon is called as inducible because this lac, the end product, so the one which the enzymes which break down the lactose, presence of the lactose will induce the expression. अगर lactose present है cell के पास, आसपास या cell के अंदर, that will induce the expression of the lac operon. That is why it is called as inducer. The lac operon, lactose, it is inducer. That is why the old system is called as like inducible system. Similarly, let's take another example of tryptophan operon. Here it is seen it as like repressible. Why it is called as repressible? If it is presence, it is switching off the gene expression, then it's repressible. क्योंकि वो repress कर रहा है. अगर if the tryptophan is present, then if the transcription is off, so what do we call such system as? We have to call it as repressible only because it is repressing the expression. Again, lactose. If the lactose is present, what is happening? The transcription is switched on. That means it is inducing the transcription. That is why it is called as inducible system. So example for the positive inducible system is CAMP mediated regulation, because here instead of the repressor protein, there are activators which are CAMP, which is binding to CRP. CAP, overall it is called as like CRP. So CAMP which binds with the CAP, and this is a protein which will activate, or these are the activators. These are collectively the activators which will activate the gene expression. That's why they are the example under positive inducible, and this is seen in the lac operon only. Lac operon mediated by CAMP regulatory protein which is called as CRP. And CRP consists of 2 important subunits which are CAMP and CAP. Okay, and negative inducible because it is negative because of the repressor and it is inducible because of the lactose. Similarly, negative repressible. It is negative because of the repressor, tryptophan operon भी repressor ही present होंगे, and it is repressible because of the presence of tryptophan. Tryptophan इसका presence repress कर रहा है, that is why it is called as like repressor. And what is about the positive repressible? There are no specific example under study. Study में कोई ऐसा specific example नहीं है. तो ये मुझे अगर मैं पूछता, like I think like most of you might have confused कि why they are called as positive regulation, why they are called as like negative regulation, why the system is called as inducible, why the system is called as repressible. I hope after watching this, if you already learned it from the master course, you will be able to recall it, or if you are watching it for first time, at least I hope you have understood what I'm trying to convey, right? Okay. Yes. Next, lac operon, it is called as inducible system, and remember it is an inducible system, but it is a negative inducible system. ये याद रखना, एग्जाम में आ सकता है. But ये सही था. Lac operon is an inducible system which can be either negatively regulated or positively regulated. Negatively regulated inducible system, the example is lac operon mediated by the repressor protein. And inducible system with the positive control, it is the one with the example is lac operon mediated by CAMP regulatory protein, which is called as CRP. Okay, so दोनों example है इसमें. Fine. Yes. Next, that means like lac operon undergoes both positive regulation and negative regulation, but whether it is positive or negative, it is constantly inducible system. इसको ये inducible system ही है, but it can be also positively regulated or negatively regulated. Okay, now let's take an example of negative regulation. Inducible तो समझ में आ गया, but let's again in inducible में दो cases थे, एक positive था, एक negative था. Let's talk about the negative. इसमें ऐसा है कि lac operon की upstream में ना एक gene होता है, what is called as lac-I gene. The protein of this gene produces a repressor. This is the one which is the repressor protein. So what is the function of the repressor? I told you the repressors have the binding site to the operator. So they go and bind to the operator. Because of the binding to the operator, the RNA polymerase cannot bypass it. The RNA polymerase cannot bypass this region. So that's why the transcription will be switched off. And this is in case of absence of lactose. Absence of lactose. Okay. Next, let's assume that lactose is present. Here the lactose is present. Here lactose present. So what happens? What difference it will make? First thing, protein repressor is like normally produced, but repressor will go and bind with the inducer. The inducer will come and bind to the repressor, and because of the binding of inducer to the repressor, this loses its binding ability to the operator. It cannot go and bind to the operator because of the inducer and inducer has come and bind it to it. What is the inducer here? Inducer is like generally इसको lactose बोलते हैं, but specifically it is like allolactose. And who converts the lactose to allolactose? कौन convert करता है? ये भी याद रख लो. So it is by beta galactosidase, which is by the lac-Z gene. lac-Z gene से जो protein आएगा ना, beta galactosidase. So that is the one which converts the lactose to allolactose and which acts as the inducer, which can go and bind to the repressors, because of which it cannot go and bind to the operator. Since it cannot go and bind to the operator, the RNA polymerase will normally bind to the promoter and it can transcribe the information, and because of which this transcription is called as switched on. Okay, it is based on the presence and absence of lactose. ये सबको पता होना चाहिए. I hope like you all know this already. Next, see in this only there is one more thing that is called as catabolite repression. ये catabolite repression मतलब lactose repression. Lactose synthesizing या breaking genes का repression. So this is the example for the positive control. See how this regulation takes place. Let me explain. Why lactose metabolism, like when the lactose metabolism is required, whenever the lactose is present in near the cell or in the cell, since to derive the energy from the lactose metabolizing genes should be expressed. So that's why lac operon will be switched on in the presence of lactose. अगर cell के पास lactose है ही नहीं, तो lac operon क्यों चाहिए होगा? So at that time the transcription will be switched off. Let's assume lactose है cell पे, but along with the lactose there is also glucose भी है. तो हमें पता है like always cell prefers the simple carbohydrate which it can easily digest, and we know the glucose is the simplest carbohydrate which can be digested, which is always prepared by the cell. So now in the presence of glucose there is a regulation that takes place. एक regulation यहाँ पे भी होता है, जब glucose present होगा then this lac operon will be switched off. ये lac operon जो है switch off होगा, क्योंकि जब glucose होगा उस टाइम पे हमें lactose का metabolism जरूरत नहीं है, क्योंकि glucose will give us the energy directly. Okay. Now whenever the glucose is absent, मान लो कि glucose absent है, so in the absence of glucose there is one more protein, the proteins which will be active, which are CRP, catabolite activating protein or catabolite regulating protein, CRP. So this will be active. This CRP, it is made up of CAMP and CAP, So this are nothing but the activators, and you know the function of activators. They go and bind to the promoter region, and because of the binding of this into the promoter region, the RNA polymerase can easily go and bind, because of which there is a transcription that takes place, and that's why the transcription will be switched on. तो याद रखना, एग्जाम में अगर पूछा जाएगा in the presence of glucose and in the absence of glucose. अगर presence रहेगा, the transcription will be switched off. How you ask me? Glucose will inhibit this protein function. जो glucose अगर present रहेगा ना, तो इसका function inhibit कर देगा. When the glucose is absent, only then this protein will be active, and it can go and activate the transcription. This is about the positive regulation by the in the lack of operon. It is mediated by CRP, which is catabolite regulatory protein. Yes. Any doubts on this? इसमें कुछ doubt है? देखो एग्जाम के लिए बहुत इंपॉर्टेंट है लैक ऑपरॉन. दैट्स व्हाई लाइक आई एम डिस्कसिंग इट इन डिटेल. तो अच्छे से याद रखना कॉन्सेप्ट को, तो एग्जाम में इंपॉर्टेंट होगा. More question from genetics, few question in general agriculture, seed science and pathology. Did not feel like I wrote the plant science paper. अच्छा, okay. Presence of repressor activators cannot act properly. Then how it is possible here? In the presence of repressors, यहाँ पे repressor कहाँ present है? इसमें repressor कहाँ है? See, in this system there is no discussion about the repressor because we are talking about the positive control. मैंने बताया था positive control it is because of the activators. हम यहाँ पे activators का बात कर रहे हैं, हम यहाँ पे repressors का बात नहीं कर रहे. And in the activators, glucose was the additional component जिसका influence से ही activators active होंगे या inactive होंगे वो decide हो रहा था. Don't club the two things. ठीक है? Okay. So next in 1966, yeah, समझ गया. I hope like you understood it. See, in the 1961, the Jacobi model proposed a model of lac operon, and in 1966 it was by Gilbert and Muller. They isolated the lac repressors. Repressors को उन्होंने completely isolate किया, and they gave the validity. एक validation दिया था, एक checking किया था, properly check करके verify किया था. हाँ, lac operon का जो भी model है ना, जो भी concept है ना, वो सही है. Operon concept बिल्कुल सही है. Okay, so that is all about the lac operon. See, one more important operon, just like lac operon, is like tryptophan operon, but it is completely opposite to it. Lac operon, it is like a catabolic process, and the tryptophan operon, it is called as anabolic process. Why? Catabolic process means it is a breakdown process. Lac operon is a breakdown process. In a very simple words, lactose is present in a cell, then there should be some enzymes which can break it and derive the energy. So that is why catabolism. Lac operon is switched on. That means the enzymes required for the breaking the lactose will be expressed. That is why entire process is called as catabolic process. Now this is anabolic or synthetic process where like synthesis takes place of amino acids tryptophan. Now if you understand this, when the lac operon genes are to be switched on, whenever there is a lactose, only then the genes should be switched on. If the lactose is not there, it should be switched off. Same way that operon was acting. Here, when the tryptophan operon should be switched on and switched off, it is just the reverse. Whenever the tryptophan is in cell, then tryptophan operon should be switched off. It should not be on because tryptophan is already in the cell. Then why there should be a gene expression which can synthesize the tryptophan, right? So whenever the tryptophan is present, the tryptophan operon should be switched off. But whenever the tryptophan operon is absent, only then this tryptophan operon should be switched on, which can synthesize this tryptophan operon. Okay? So that's what the simple logic. If you understand it, you can easily answer any type of questions. So this tryptophan operon, it is regulated by the two different methods. One is like it involves the end product. What is the end product? It is tryptophan. Tryptophan concentration से ही regulate हो जाता है. First step. In the second step, it is by attenuation mechanism. ये नया term होगा. Attenuation mechanism which acts in the tryptophan operon. There are two possibilities of how the tryptophan is regulated. One is by the end product, second one is by the attenuation mechanism. So let's understand both of it. First one, tryptophan operon, it is an example for negative repressible. Why repressible? Because the tryptophan in the presence will repress the operon. Now, understand this. there is a regulatory gene which will give the repressor. यहाँ पे भी एक repressor produce हो रहा है, but this repressor is like inactive. ये inherently inactive होगा. Okay, it becomes only active when there is a co-repressor that is there. So which is acting as a co-repressor here? It is tryptophan. Tryptophan रहेगा तो ये repressor को activate करेगा, and if the repressor gets activated, it can go and bind to the repressor, and because of which the transcription will be switched off. But in the other case, if the cell does not have the tryptophan है ही नहीं, तो जो repressor produce हुआ है that will be like inactive. ये inactive रहेगा. If it is inactive, it cannot go and bind to the operator region. Because of it cannot go and bind to the operator region. The RNA polymerase will normally go and bind to the promoter, and because of which there will be synthesis of all the genes that takes place. And remember, there are 5 genes in the tryptophan operon, and there are 3 genes in the LAC operon. LAC operon में 3 genes थे, which is like LAC, Z, Y, A, and in case of tryptophan operon, there are 5 which are like in the alphabetical order E, A, B, C, D, E. So there are 5 genes which are present in the tryptophan operon. Okay. Next, so involves a secondary structure, and it is called the attenuation mechanisms. So depending on the secondary structure of the kind of structure, depending on the structure the mRNA is making, that will decide whether transcription will takes place or will continue or it will terminate. So that is why it is called as it involves a secondary structure. So whenever this mRNA was tryptophan mRNA was studied in detail, they found they found some structure to be like very different. So what is the difference? See, entire structure if they studied, it has some peptide which is called as leader peptide. There are some sequence in the there is some sequences between this region in case of tryptophan which codes for some peptide gene. So this has revealed that it has some structural differences. So what is the structural differences? And it is called as leader transcript. इसको leader transcript बोलते हैं. And it has structural differences where there are 4 different regions. It can be divided into 4 different regions. जहाँ पे first region and second regions are complementary each other, which can make a bind, like which can bind to each other, or which can make or pair with each other. Next, third and fourth are complementary to each other, which can again make the pair, or the 2 and 3 again they are complementary each other, which can make a pair between them. तो यहाँ पे 2 possibility है structure का. What are the 2 possibilities? What are the 2 possibilities? The 2 possibilities are either the loop can form between 1 and 2. अगर एक के साथ 2 loop form कर रहा है, then there will be loop formation between 3 and 4. ये एक possibility हो गया. अगर 2 and 3 are forming the loop, they are forming the pairing. अगर वो bind हो रहा है, so साथ में आके bonding हो रहा है, then the other 2 cannot. So there can be either loop between 1, 2 and 3, 4, or there can be loop between 2 and 3. So based on this difference only, the tryptophan operon is controlled, the regulation takes place. So how, if you want to understand in a simple words, इतना याद रखना, जब अगर excess tryptophan होगा सेल के अंदर, तो ये एक और भी चीज यहाँ पे डिस्कवर हुआ था, that this leader peptide has the sequence of this, जहां पे स्टार्टिंग में there are 2 adjacent tryptophan. ये लीडर पेप्टाइड में 2 adjacent tryptophan है, अमीनो एसिड है जो कोडिंग सीक्वेंस में है. ठीक है, दिस इज द mRNA. mRNA के बाद जब अमीनो एसिड फॉर्म होगा, that अमीनो एसिड चेन had 2 tryptophan which are adjacent to that, adjacent to each other. इसको याद रखना, ये इंपॉर्टेंट है. So what happens whenever there is a less tryptophan है ही नहीं सेल के अंदर. If the tryptophan are very less, now tell me, if this is DNA, the mRNA is coming out, यहाँ पे leader peptide first transcribe होगा, and then other regions are getting transcribed. So now when this leader peptide is getting transcribed, since like tryptophan are not there, so tRNA which brings the tryptophan which will make a delay, थोड़ा delay करेगा वो. ठीक है. So because of this delay, so where does the loop formation takes place? यहाँ पे loop कहाँ पे form होगा? फर्स्ट केस मैंने फर्स्ट केस थोड़ा उल्टा बताया, इसको भूल जाओ. लेट्स से लाइक ट्रिप्टोफैन इज नॉट देयर इन द सेल, सेल के अंदर ट्रिप्टोफैन है ही नहीं, ठीक है। जब स्टार्टिंग में ट्रांसलेशन होने के लिए टीआरएनए को ट्रिप्टोफैन उठा के ले आना पड़ेगा, दैट मींस लाइक चार्जिंग ऑफ टीआरएनए करना पड़ेगा। सो सिंस लाइक दैट tryptophan is not present in the cell, so it will take much time to take the tRNA there charging, so because of which a region तो ribosome catch कर लेगा but it cannot overlap between two regions, so because of that there is a loop formation between 2 to 3 regions, 2 and 3 second and third region में loop form कर देगा, and this loop is a row dependent terminator, that means this loop needs row factor to terminate the entire process. Since this is a row dependent terminator, it will not terminate the process, because of this the transcription will be switched on, ये switch on रहेगा whenever there is a tryptophan starvation. but मान लो कि excess tryptophan है सेल के अंदर, तो immediately जैसा ही mRNA बाहर आ रहा है, the tRNA will go and give the tryptophan, because of which 1 and 2 region जो leader peptide के होंगे, it is already occupied by the ribosomal complex. So loop formation can only takes place between the 3 and 4, and because the 3 and 4 region is a row independent terminator, it is a row independent terminator, row चाहिए नहीं होगा इसको terminate करने के लिए process को, so it can easily terminate, and because of this the transcription will be switched off in case of excess of tryptophan. So that is what you have to understand between tryptophan presence and absence, या logically जैसा मैंने बताया ऐसा भी याद कर सकते हो, अगर सेल के अंदर ऑलरेडी ट्रिप्टोफैन है तो ट्रिप्टोफैन ऊपर ऑन होने का जरूरत है, ऑलरेडी सेल के अंदर ट्रिप्टोफैन है, देन इज देयर नीड टू सिंथेसाइज द जीन व्हिच विल गिव अस द ट्रिप्टोफैन? बिल्कुल नहीं, क्योंकि ऑलरेडी ट्रिप्टोफैन है सेल के अंदर, तो फिर से जीन एक्सप्रेशन क्यों करना पड़ेगा ट्रिप्टोफैन सिंथेसिस करने के लिए? बट इफ द ट्रिप्टोफैन इज नॉट प्रेजेंट इन द सेल, देन इज देयर एनी नीड फॉर gene expression of tryptophan operon? Yes, there is a need, because cell के अंदर tryptophan नहीं है and this amino acid is required, so that's why the transcription will be switched on. ठीक है, so that is what you have to remember. Next, so this is all about the regulation in the prokaryotes. Prokaryotic regulation जब बात करेंगे, like you have to know about the prokaryotes में activators के बारे में पता होना चाहिए, like you have to know about the repressors and you have to know about the positive regulation and the negative regulation. Next, the lac operon, which is mediated by the lac repressor and it is mediated by the catabolite repression, which is by the positive control CMP mediated control. Next, the tryptophan operon, which is again controlled by the two different processes. First is by the end product, second is by forming the secondary structure. So this is what is important for the regulation of gene expression in the prokaryotes. Next, there is a regulation of gene expression in case of eukaryotes. So regulation of gene expression in the eukaryotes, there are different examples of regulation. Some of the very important examples you can study is like position effect. In that position effect, again there are 3 categories. One is variegation effect, cis and trans effect, and duplication effect. So what is position effect? Based on the position of a gene inside the genome, the expression will decide whether into which location or to which region the gene is present. That will decide its expression, whether it will be expressed or not. So one important example is variegated position effect. So this is seen in mottled eye mutants in case of Drosophila. अगर ये chromosome है, ठीक है, and if there is a gene that is present here, let's assume it is like A gene, and if this A gene is getting inverted, अगर invert होके near heterochromatin आ रहा है, then you know that you know the process of heterochromatinization. Whenever the regions that are close to the heterochromatin, that can be heterochromatinized. Because of this, the gene expression changes. This is how the regulation happens. This is the one example in the variegated position effect. Next, cis transposition effect. Based on the cis and transposition, जैसे कि इसको क्या बोलेंगे? This is transposition. In the transposition, it is giving the apricot eye color. Next, if it is in the if this is in the cis formation, same combination, this is in the trans and this is in the cis, it is giving the dull color. See, just because of the cis and trans positions, even though the gene combination is same, even though allele combination is same, it's still changing the expression. That is what the cis and trans effect means. Next, the duplication position effect. Duplication position effect, because of the unequal crossing over, we have seen in the bar eye, because of the multiplication of the 16A region, this phenotype itself changes. The example is bar eye gene. So that's what you can remember where the regulation takes place. Next, this is a one type of regulation because of the position effect. Next is epigenetic regulation or epigenetic inheritance. This is also a change in a जीन एक्सप्रेशन, एपिजेनेटिक का मीनिंग या डेफिनेशन अच्छे से समझ लो. दिस इज अ चेंज इन अ जीन एक्सप्रेशन विदाउट अल्टरिंग द डीएनए सीक्वेंस. यहाँ पे डीएनए सीक्वेंस कुछ अल्टर नहीं होंगे, नो म्यूटेशन, नो क्रोमोसोमल एबरेशंस, बट देयर विल बी चेंज इन द जीन एक्सप्रेशन एंड इट इज बिकॉज ऑफ जनरली द मिथाइलेशन. अगर डीएनए मिथाइलेशन से ही ये मेजरली होता है, चेंज इन द मिथाइलेशन स्टेट, सो देयर इज अ चेंज इन एक्सप्रेशन. ठीक है, इसको याद रखो. And the third one, the genetic imprinting. So genetic imprinting is like marking. So based on whether it is like whether it is transferred from mother or the father or the mother, it will be decided whether it will be expressed or not. Some genes are paternal in expression, that if they are coming from the father, only they will be expressed. Whatever alleles that are coming from the mother, they will be not be expressed. And there will be some maternally expressed genes where if they are coming from the mother, they will express. If they are coming from the father, they will not be expressing. That is what genomic or genetic imprinting means. This is again based on the DNA methylation state. So DNA methylation state will decide whether the genes will be expressed or not. Okay? Yes. Next. So again, similarly, just like a prokaryotes, eukaryotes may be transcription different level of regulations hai. कुछ different level of regulations है. So first one is like transcription level है. Transcription level पे regulation जब होगा, so it is like of different. This is like post transcription. ये जो है ना, ये post transcription है. Transcription level पे just what we have seen in the prokaryotes, eukaryotes may be either by the change in the chromatin structure which can influence the transcription process or overall change in the genome structure. So the regulation takes place at a transcription level. ठीक है, generally बता रहा हूँ, ज़्यादा depth में कुछ भी है नहीं. There is nothing but one thing to understand. Transcription level पे कैसे regulation possible है in case of eukaryotes? It is by changing the structure of the chromatin. अगर chromatin structure में कुछ भी ऊपर नीचे, nucleosome structure में, overall chromatin structure में या chromosome structure में कुछ भी alter कर दिया, then that will definitely affect or directly affect the transcription. So that is what you can understand. And here why I have given this is, at a transcription level, the chromatin, there are two important models, nucleosome remodeling or histone chromatin change. So histone chromatin change model में, these are the process that can take place which can change the structure of the histone, like histone covalent histone modification. We say it as like covalent histone modification. हिस्टोन के स्ट्रक्चर में या क्रोमेटिन स्ट्रक्चर में चेंजेस होने के वजह से रेगुलेशन हो सकता है, एंड दिस इज द पॉसिबल mechanisms of how the covalent histone modification can takes place. And out of this, if you have heard about acetylation, methylation and phosphorylation, these are the most common types of changes or the mechanism that takes place which will change the structure of chromatin or the covalent histones, which are the core integral structure of the chromatin. ठीक है, difference between the paramutation and the epigenetics. Paramutation is one of the type under the epigenetics. Epigenetic का एक type है. Class bore हो रहा है क्या? Class is getting bored. Sir, regulation part आते आते ही दिमाग खराब हो रहा है. तो इसको इतना अच्छे से पढ़ा नहीं हमने. जितना सिंप्लीफाई हो सके उतना सिंप्लीफाइड वे में बता रहा हूँ, इससे ज्यादा कुछ है नहीं. और 2-3 स्लाइड है, that will complete the regulation process. Next we have one more important topic, that is transposable elements. I'll take another 20 minutes, that will be also completed. वो भी complete हो जाएगा. ठीक है, so don't lose your concentration, अच्छे से देख लेना. Okay, next, that was regarding the transcriptional level. Now what about the post transcription level? Post transcription level पे भी regulation possible, and there are again two important examples here. Transcription होने के बाद भी during the post transcriptional, that is like at a translational level. This is at a translational level. So translation level पे, just like in the lac-operon, we have a positive and negative regulation. Similarly, eukaryotes में एक example है which is like negative and the positive regulation. Negative regulation in case of ferritin synthesis genes, and the positive regulation in case of ferritin. There is one more, transferrin synthesis genes. Okay, first one is like ferritin synthesis and next one is like transferrin receptor synthesis. This is an example of negative regulation. Example याद करना, ferritin synthesis is the example for negative regulation and transferrin receptor synthesis, it is an example for positive regulation. Okay, इतना ही याद कर लेना, इससे डिटेल में नहीं जाएंगे, स्किप कर लेंगे. ये पॉजिटिव रेगुलेशन है. अच्छा, दोनों में नेगेटिव दिया क्या मैंने? ओके, सो दिस फिगर विल बी लाइक चेंज्ड. यहाँ पे एक पॉजिटिव रेगुलेशन का एग्जांपल आएगा. सो व्हाट इज द एग्जांपल फॉर द पॉजिटिव रेगुलेशन एट ट्रांसलेशन लेवल? इट इज transferrin receptor transferrin receptor synthesis, इसमें positive regulation होता है and ferritin synthesis में there is a negative regulation. Okay, so now what we have understood from the eukaryotes, इतना तो समझ सकते हो आप लोग. Prokaryotes में हमने क्या समझा था? Prokaryotes में regulation mainly किस वजह से हो रहा था? It is because of proteins. But eukaryotes में थोड़ा complex है. एक तो proteins था ही which will regulate the process of gene expression, but along with the proteins there are also the RNAs which can directly regulate the gene expression. यहाँ पे RNAs भी आ जाते हैं पिक्चर में जो regulate करते हैं gene expression को, and this is called as like RNA interference or RNA technology. So in this, didn't drink mango juice. Mango juice नहीं था वो orange energy drink था. Sir, genomic imprinting is present in the animals and humans. Yes, it is present. Genomic in the humans, like you take the example of bar body, where it is like entire one genome is imprinted, one copy of the genome is imprinted. That is also an example of genomic imprinting or genetic imprinting. Okay. Okay. So this is as just like the proteins are involved, which can be like activators or the repressors in the regulation of gene expression. Here, the RNAs are also involved in the regulation of gene expression. So one very good example is about RNA interference technology. And for the first time, it is by Fire and Milo, yaad rakhna, first established this in the C. elegans, RNA technology. And this is also called as post-transcriptional gene silencing. Transcription होने के बाद भी अगर gene silence कर रहे, so that is what this is called as post-transcriptional gene silencing or transgene silencing or quelling, other names. And there are two types of RNAs which are involved in the silencing, microRNA and small interferon RNA. So there are two types of RNA, miRNA and siRNA, which are involved in the regulation process, which are small silencing RNAs, which are involved in the regulation process. Next, post translation. ठीक है, हमने transcription level पे देख लिया, translation level पे देख लिया. Next, even after like translation also, even after the proteins have been formed, the polypeptides which are formed, again they can undergo modification, and this modification can result in the regulations. So these are some of the process that can lead into the amino acid modifications. As such इसको याद नहीं रखना होगा, but एक बार देख लेना बस. कुछ important नहीं है इसमें, सिर्फ समझने के लिए देख लेना कि proteins produce होने के बाद भी there is a possibility of regulation by changing or modifying the amino acids which are formed. That is what comes under like post translational modification. Okay, so this is all about the regulation process. Regulation आपको सबको समझ में आया होगा. Like if I hope like everyone has understood the regulation nicely. जितना brief में हो सकता था उतना brief में मैंने बताया है, but still इसके अलावा भी कुछ भी doubts पड़ता है तो you can comment it on the video. So इसके बाद में जब video class के बाद भी अगर जब भी मैं देखूंगा I will be answering it. ठीक है? Okay. जितना स्ट्रेंथ देख रहा हूँ देखने से ही लग रहा है कि आप लोगों में रेगुलेशन पार्ट में इंपॉर्टेंट नहीं है, बट लेट मी टेल यू रेगुलेशन से क्वेश्चंस आएंगे. तो अगर क्वेश्चंस आ रहा है लाइक यू विल लूज इट इफ यू डोंट अंडरस्टैंड इट. ओके, सो द लास्ट पार्ट इज लाइक ट्रांसपोजेबल एलिमेंट्स. दैट इज वन ऑफ द वेरी इंपॉर्टेंट पार्ट, क्योंकि यहाँ से भी डायरेक्ट क्वेश्चंस आ जाते हैं. सी व्हाट आर ट्रांसपोजेबल एलिमेंट्स? ट्रांसपोजेबल एलिमेंट्स आर नथिंग बट द डीएनए सीक्वेंसेस. ये डीएनए सीक्वेंसेस ही है, but they have like unique capability that they can transfer from one position to another position. For example, if this is a DNA and if I say this is a transposable elements, so it can cut itself from this position and go and attach to any other place, or it can copy itself. Cut हो सकता है या copy कर सकते हैं itself and then again go and attach to the new regions. It can multiply in the copies or cut itself and go in anywhere in the same genome. So that is what the transposable elements means. That means it can move across within a genome. They are only present in all types of organisms. एक common है, हर एक organisms में transposable element common है. And in the humans there are 40%. 40% of the genome contains like 40% of genome is made up of transposable elements. And this transposable elements, for ease of clarity and understanding, can be divided into simply three different types. One is like cut paste transposons, replicated transposons, and third one is like retro transposons. So you ask me like, so what are these? So these are the three different types. Cut paste transposons are nothing but whenever they want to transpose, they will cut from their initial position and then they go to the another position. So whatever transposons that are of this nature, they all come under this cut paste transposons. जहाँ पे original position वो cut होंगे और नया position में जाके paste होंगे. Next, replicated transposons, they are the one जहां पे एक अगर ये region है, यहाँ से transposable element है, they will copy itself, then this copied will be going and placing it to the new location, जहां पे original location में भी है और नया location में भी जाएगा. So that is called as copy paste or replicated transposons. Third one, retrotransposons. Retrotransposons is a name because if this is a DNA segments, it will first make the RNA, that is like mRNA, and then with this, like there is a cDNA which is formed, and this cDNA, if it is going and integrating, which is the mechanism of retroviruses, or the one which uses the reverse transcriptase, since they are using this mechanism, this category is using this mechanisms, all such transposons are called as like retrotransposons. And again, in the retrotransposons, there are two different types. One is retrovirus-like elements, second one is like retroposons. So what is the difference between these two? Retrovirus-like elements are those transposable elements which are similar to the retrovirus. Retrovirus, agar if you want to understand what are retroviruses are the very simple virus which is having like only genetic material, and this genetic material have a genes for three important genes, which is like GAG, ENV and polymerase, ENV and GAG. So these are the main genes that are present in all retroviruses. जितने भी retroviruses है ना, उसके पास ये तीन genes है. So this transposable elements which are there in the different organisms, they match to these genes. जो transposable elements है ना, जो sequence है, they match to the information present in the GAG and pol. ENV छोड़ के बाकी दो genes का they are homologous. The transposable elements are homologous under this category. That is why they are called as retrovirus-like elements. याद रखना, ENV का homologous नहीं है, but GAG और pol का homologous present होंगे. So that is what retrovirus-like elements means. ठीक है? Yes. Next, retroposons. Retrovirus-like ही है क्योंकि they have this mechanism of DNA to mRNA, then to cDNA and then to DNA. So that is why they are under the category of retrotransposons and they are called as retroposons. So इसमें आपको क्या-क्या याद करना होगा मैं इसी स्लाइड में बता देता हूँ. I will not go ahead of this slide. इस स्लाइड से आगे नहीं जाऊंगा. यहीं पे एग्जांपल बता देता हूँ. एग्जाम के लिए क्या इंपॉर्टेंट है? You have to remember the examples of cut paste mechanisms. Cut paste में जितना भी एग्जांपल्स यहाँ पे डिफाइंड है ना, और कौन-कौन से ऑर्गेनिजम्स में है, ये एज इट इज आपको याद करने होंगे. Most important is like ACEs and DS. Why it is most important? You know, like most of the phenomena is like discovered in the prokaryotes, but the transposable elements is the one thing which is primarily discovered in the eukaryotes, and it was by McClintock, and it was this same AC and DS activators and dissociators elements that was seen in the maize genome, which was responsible for stripping and spotting in case of maize, right? And apart from that, there are P elements which are important. There are IS elements which are seen in the bacteria, and there is composite transposons they are found in the bacteria. There are PGB, Obo elements, Sleeping Beauty, they are present in the salmon, moth and Drosophila. So these are all the examples, these are all the representative examples in case of cut paste transposons जो exam में directly आ सकता है, तो याद रखना. Next, the examples for the replicative transposons, copy paste transposons are TN3 family elements which are commonly called as TN3 like elements. ठीक है. Next, so what are the example for retrotransposons which will utilize the RNA as an intermediate and then transpose? So they are divided into two different types, as I told you, like retrovirus like elements and retroposons. So these are called as like LTR. LTR means like they have a long terminal repeats. End पे उसके पास long terminal repeats होंगे. That is why the name LTRs. And the retroposons, they are called as non-LTRs because they don't have the long terminal repeats. Examples for this LTRs are TY1, Kupi and Gypsy. And examples for non-LTRs, this is in the humans also. Remember, this is Lins and Sins. Lins का main example है L1 and Sins का main example है, it is Alu. And the Lins are the one which are active, or we call it as like autonomous. इसको हम बोल सकते हैं कि these are autonomous and these are non-autonomous. And autonomous मतलब by on their own they have the ability to transpose from one position to another position. But non-autonomous means they do not have the inherent ability to transpose. They need the autonomous elements together to be present in the genome to get transposed to the different region. Similarly, in the ACs and DS also, you know, ACs are the one which are autonomous. इसको autonomous इसलिए बोलते हैं क्योंकि they are capable of transposing to one area to or one region to another region in the genome. Dissociators, they don't have they are non-autonomous because they don't have the inherent ability where they can transpose on its their own. ये याद रखना, ठीक है? Next, so examples. What are sins and lanes? Definition इसका full form भी दिया हूँ. Long interspersed nuclear elements. Exam में आ सकता है. Lins, the full form is long interspersed nuclear elements. Sins, short interspersed nuclear elements. ये दोनों humans पे present है, इसलिए important बोला मैंने. Lins and sins are present in the humans, in which lins are the active one. The major example in the lins is L1. In the sins, there are alu elements, which are the major examples. Okay, so from this directly questions can come in the exam. So make sure that you remember this part. So along with that, like I have also added few other things. Like in the bacteria or in the prokaryotes, you can divide the transposable elements into 3 different types. See, transposable elements can be divided into 3 different types in case of bacteria. First one is IS elements, second one is composite transposons, third one is like TN3 elements. What is the difference between these 3? Insertion sequence are the one which are the simple transposable elements which has a region for the transposase gene which is responsible for the transposition, and the ends have the inverted repeats. They have inverted repeats, as you can see, they have inverted repeats. So that is what IS sequence means. So what is composite transposons? So this is a composite transposon. Example is TN9. If one insertion sequence is inserted into the another insertion sequence, that is called the composite transpose. जहाँ पे देखो एक insertion sequence end पे present है. The insertion sequence are present on the ends of the another elements. So they are called as composite transposons. The example is TN9. जहाँ पे एक पूरा stretch of DNA में ends पे insertion sequence आके बैठ गया है, तो इसका पूरा structure को हम बोलेंगे composite transposons. The example is TN9. Next we have TN3 like elements. So these are the one primary examples जहाँ पे copy paste mechanism से ये transpose होते हैं, and this too, the insertion sequence and the composite transpose, remember, they transpose to the copy paste mechanism. So that is the difference between these 3. ठीक है? And this TN3 like elements, one example is TN3. TN3 like elements क्यों बोलते हैं? क्योंकि this is a broader term, a family of transposons which also includes TN3, which is an important transposable element. And this is the example given. The figure is about the TN3 only. This has the genes, 3 important genes. One is like transposase, one is like resolvase, third one is like beta lactamase. So these are the 3 important genes that are present in the TN3 element. And how it is different from the other 2? This is different because this is copy paste mechanism, or it utilizes the copy paste mechanism. The other 2 utilizes the cut paste mechanism. This is what you have to remember, and all 3 are present in the prokaryote. We are talking about the bacterial transposable elements. If they in the exam, if they ask you about the examples for the bacterial transposable elements, so these are the 3 that you have to remember. Okay? And how that transposition takes place, and you also have to know transposable elements, if they are transposing from one location to another location, there is a target site duplication that can take place. How? Let's take an example. Let's take the insertion sequence which are attaching to the new region. So this is a new DNA region. There is a random cut that is first made, and once the random cut is made, this is the insertion sequence which is coming and attaching. Because of the insertion of the insertion sequence, because of the addition of this insertion sequence, you can see this leftover part, it is again synthesized. Because of this synthesis, you can see exactly that these regions have been duplicated. Earlier it was in single copy. Because of this transposition, it has come into 2 copies. So this is why we say whenever there is a transpositions, there is a target site duplication that takes place. Okay. And in case of eukaryotes का एक main example जो था AC and DS था. इसका मैं बता दूं, like you will understand this very nicely. ACs are activators. Why they are activators? Because this is a structure of AC. It has a complete information that is present, nothing is deleted, and it can easily transpose itself from one region to another region within a genome. Next, there is a DS elements which are derived from the AC only. AC is elements where there is a deletion in some part, that is called as DS. DS are actually AC only. Activators का ही genes same sequence है, but कुछ रीजंस उसमें डिलीटेड है, बिकॉज ऑफ दिस डिलीशन ना दे डोंट हैव एबिलिटी टू ट्रांसपोज देमसेल्फ, बिकॉज ट्रांसपोजन जीन इसी के अंदर होता है, वो ट्रांसलेट होके प्रोटीन नहीं फॉर्म कर सकता, एंजाइम नहीं फॉर्म कर सकता, बिकॉज ऑफ दैट it cannot transposition or it cannot transpose from one position to another position. That's why they are called as DS elements. They are nothing but the AC only, but with some major deletion. Next, other than that, there is like aberrant DS elements. Aberrant DS elements is where DS elements है, but उसके अंदर अगर कोई other DNA आके बीच में है, if some other DNA it is been added to that DS element, that is called as like aberrant DS element. जो इसके सीक्वेंस सिमिलर नहीं है, जो नॉन होमोलॉगस डीएनए है, कुछ अदर पार्ट्स के डीएनए आके बीच में इन मिडल इफ इट गेट अटैच्ड to the DS elements, those are called as like aberrant DS elements. Next, double DS elements. If one stretch of DS elements, in between that another stretch of DS elements come and bind to it or attach to it, that is called as like double DS elements. See, all three, 1, 2, 3 and 4, out of this only one has the ability to transpose itself autonomously. That is why it is called as like autonomous elements. And the other three, these three, they cannot transpose by themselves. They need the activator genes or the protein product of the ACs to transpose themselves. That is why they are called as like non-autonomous. This is about the ACs and DS discovered by McClintock. Okay, so last one is whatever I said, it is about the retrovirus and retroposons. Retrovirus, why the name retrovirus? Because they have a genes which are homologous to the retrovirus genes, which are GAG and POL. Okay, two genes are homologous in case of transposable elements and the retrovirus. That is why they are called as like retrovirus like elements, and they are also called as LTR transposons because they have long terminal repeats. Other one is like non-LTRs, that is retroposons. They are non-LTRs because they don't have long terminals repeats, and these are the examples. So this is what you have to read for your exam. So यही है जो आपके एग्जाम के लिए important है. You and your team is doing great work. Your hard work and effort is very appreciated. I have a humble request, please upload more videos on one shot of horticulture. अच्छा ठीक है, horticulture को तो already marathons हो गए ना, तो how much videos we want? Are you asking for like ICAR PG 2026 or for like later year, or if you are asking for any other ICAR PhD or any other exams or ASRB, anything? So guys, so this is all about the today's class. So complete marathon complete कर दिया हमने molecular genetics का. हालांकि थोड़ा boring लगा होगा. I did not wanted to bore you people, but यहाँ का audience का ऊपर नीचे इतना हो गया, मुझे लग रहा था कि कुछ लोगों को तो बिल्कुल दिमाग के ऊपर से जा रहा है ये चीज. But still, देखो मैंने clearly बताया कि एग्जाम में क्या पूछेंगे क्या नहीं पूछेंगे. Like I have clearly told like what question comes in the exam. So please ensure कि you remember them as such. अगर समझोगे नहीं समझोगे सेकेंडरी है, बट एटलीस्ट याद तो कर लो यार. तो एग्जाम में आएगा तो आंसर तो करना चाहिए ना. रिवाइज इट, प्लीज रिवाइज इट. सर थैंक यू सो मच, प्लीज रिवाइज इट. रिवाइज हो गया, अच्छा. When will the admission come? When will the admission come मतलब admit card. admit कब आएंगे सर? admit card will come in next 2 to 3 days. 4 जुलाई को एग्जाम है, तो 2 जुलाई आज या कल में आ ही जाएगा. क्या पता है कि आज ही आ जाए मिडनाइट पे या कल सुबह आ जाए, एनी टाइम आ जाएगा. ऑलरेडी सिटी इंटीमेशन हो गया ना, तो कोई टेंशन नहीं है. एडमिट कार्ड एक दिन पहले भी आ जाएगा तो क्या फर्क पड़ रहा है, राइट? सो विल बी ऑल प्रिपेयर होंगे. खाना खा लेना. थैंक यू ब्रो. खाना तो मैंने आज खा के आया था एक्चुअली. आफ्टरनून में लंच स्किप कर दिया था, तो आई हैड टू ईट, अदरवाइज देयर वुड बी नो एनर्जी. इसलिए भागते भागते खाना खा के आया. Guys, like if you like the session, just drop a heart. And this is like we are officially closing this genetics part. Genetics को हमने कितना मैराथन में खत्म किया? So there are 3 for Mendelian, 3 for Mendelian, and 2 for molecular, and 1 for population. Mendelian general history थे के how much हो गया? 1, 2, 3, 4, 6 sessions for genetics. जेनेटिक्स को हमने 6 सेशन में खत्म किया है. टोटल आई गेस 6 सेशन मतलब 12 घंटा, लाइक ऑलमोस्ट लाइक 12 आवर्स. very videos of marathon and horticulture ICAR PG. No, there are videos, but they are in the Hindi medium. Like you might have missed it. Just drop a WhatsApp message on this number. We will share you the YouTube link, YouTube video link. ठीक है, YouTube video link share करेंगे. Okay guys, so that's it from my end. मेरा energy गया. Like energy तो है, इतना जल्दी नहीं गया, but still पोर्शन खत्म हो गया जेनेटिक्स का. So I'll come back tomorrow with the seed science. जो आप seed science बोल रहे थे, उसका एक ही सेशन में बहुत ही कॉम्प्रहेंसिवली, बहुत ही सिंप्लीफाई करके क्लैरिटी के साथ I'll discuss all the relevant things which are important for your exam. So that completes the marathon series from AgriAddict for the plant sciences. I'll study. You are the best. Thank you so much for all the love. What should be the next 2 days plan? Any suggestions? Suggestions कुछ भी नहीं है, थोड़ा काम रहो, ज्यादा हरी भरी मत करो, जो पढ़ा है उसको आराम से टाइम लेके पढ़ते जाना. बट नेक्स्ट 2 डेज में, नेक्स्ट 3 डेज मान लो कि आपके पास 3 डेज है, मेक श्योर कि नेक्स्ट 3 डेज में ऊपर से सारे सब्जेक्ट्स को एक बार रिवाइज कर लो. ये स्ट्रेटजी बना देना कि हाँ, फर्स्ट हाफ में मॉर्निंग से आफ्टरनून तक ये सब्जेक्ट करूंगा, आफ्टरनून से शाम तक ये सब्जेक्ट करूंगा. सो दैट्स हाउ इट विल डू इट. एंड बीच में मॉक टेस्ट अटेंड करना होगा तो अटेंड कर लेना, बट आई डोंट रिकमेंड यू कि थर्ड के बाद कुछ भी अटेंड करना. वॉट एवर यू अटेंड ना, कीप इट टिल द 2nd of May, 2nd of July. 2nd of July के बाद 3rd of July को कुछ भी अटेंड मत करना. सिंपली जो स्टैट्स होंगे, जो मेमोरी बेस्ड कंटेंट होगा, उसको रिवाइज करते जाओ. बस ज्यादा डिस्कशन में भी इन्वॉल्व मत हो जाओ. एंड ऑल्सो डोंट वरी अबाउट द कॉन्सेप्ट्स इफ यू आर नॉट एबल टू recall. If there are simple concepts, try to revise it. If there are complex things, don't just scratch your head out. Do not start watching the videos again. Don't do it at the last day. Today no more class. Good night. Today no more class. No more class. हो गया ना आज का, तो कल करेंगे ना. So we'll do it. Seed science का चाहिए, like everyone wants seed science, or shall we drop it? Drop कर देंगे. जो मास्टर क्लास में पढ़ाया था वही काफी है. अच्छा से पढ़ाया होगा ना मैंने. So I dealt it nicely ना. So shall we drop it or we have to revise it again? Sir, we will meet in the Noida. Who is this? Abhinav. अच्छा, okay. Okay, I'll be very happy to meet. एक बार मैसेज कर देना. See, whoever will be coming to the Noida city center, you can please message me on this number so that I will be able to coordinate with you, contact you, and we will be able to meet even at least before the exam or after the exam. Anything is fine. I'll be also there near the exam center at least like 30-40 minutes after the exam. Can't drop. Want. We need. Please don't get it done today itself. Get it done today itself. काम पूरा रात भर करेंगे क्या? पहले तो पीपीटी तो मैं तैयार करने दो भाई. पीपीटी अभी रेडी नहीं है, पीडीएफ रेडी नहीं है. Of course we want the seed science marathon. Yes, we will do it. ठीक है. And हाँ, like if you are coming to Noida, like if your city center or exam center is in Noida, please drop a message on this number, and I will be very happy to coordinate and connect with you and meet you on the exam day. Even if you are like very much tense of the exam के बाद मिल लेंगे. ऐसा कंपल्सन नहीं कि एग्जाम से पहले ही मिलना है मुझसे. एग्जाम के बाद भी मिलोगे तो कोई प्रॉब्लम नहीं है. टुडे इवनिंग. अच्छा, कल. अरे भाई, कल का मतलब वी आर गोइंग टू डू इट इन द 1st of July ओनली. आज ही, ठीक है, आज ही करेंगे. सर कम टू जबलपुर. आएंगे नेक्स्ट ईयर. लाइक वी आर प्लानिंग टू विजिट ऑल द agriculture universities. Definitely will come. Hope like everything is clear from the genetics. जितना possible था मेरे end से मैंने push किया. It was not easy even for you and even for me. पढ़ाना easy है, but last moment में जब बहुत सारा काम होता है तो उसके बीच में time निकाल के ये करना थोड़ा मुश्किल है. But still I managed to do it, and of course like you people have supported, you people are very patient कि आप लोग हमेशा मेरे क्लास के लिए वेट किए हो. So I'm very much grateful for that कि जितना आप लोगों ने प्यार दिया, सपोर्ट किया, and what you have showed all the love and patience towards my classes. Thank you so much for that. Okay, we'll meet you in the next class. MP, yes, we'll also come to MP. Okay, meet you in the next class. Good night everyone. Have a nice sleep.